some science efforts that are occurring at Ocean Sciences.
Oh, sorry.
Yeah, so I think that will, including that those transects. You said, can you remind me how long that was going to take in total? Like about?
So it should be about 70 minutes total
70 minutes.
for each of the transects. Of course, there's the ascension time between each, so we may add more than an hour to accommodate these. But there are 7 total transects, 10 minutes each, and the depth ranges are 2,000 meters, 1,500 meters, 1,200 meters, 900 meters, 700 meters, 500 meters, and whatever other scattering layer we identify in the EK60 data, which is often above 500.
Okay, so it sounds like, you know, maybe close to 2 hours or an hour and 40 minutes for those transects.
We dredged along a line.
So if we
3
just
kilometers from here to here.
if we can extend the dive by 2 hours, that will be the 2 hours that will be dedicated to the transect. We still have 8 hours minus an hour and 20 minutes in each side, so that's 2 hours and 40 minutes. So we should have
5 hours 20.
5 hours 20 of bottom time. Okay, yeah. So with the current track that we're looking at was around 500 meters in distance.
It was 550.
You just turned them off.
550. So maybe we can shorten it a little bit, but keep it around 500 meters and just in a similar shape what it is now. And I think if everybody agrees, we can save this as a tentative waypoints, and we can get started on today's dive. We're seeing the seafloor right now. Perhaps if you want to continue the discussions, we can do another. Or continue this dive meeting as soon as we leave the seafloor today and finalize or refine any further points that you might want to
So Matt, I'm looking at this rock with the lasers on it.
Yep.
That look good?
I think that's a good idea.
Sounds great. So everybody's happy with this, is that right? I'm not sure you guys, with Santiago, Matt, you both are okay with that compromise? Or?
Yeah, as I was saying, I think I agree with all the points that everybody raised. I think I like, as similar to what Tim suggested, I like to see a diversity of depths, but I also want to spend
That's pretty cemented in there.
time on the top of the ridge of the Seamount. We haven't been able to do that so far, and I think it will be one of the places where I would expect to see, as you mentioned, the highest abundance in coral and sponge communities on top of there. So I think a track like the one we just discussed and Drew will likely accomplish all these objectives.
And Matt's okay with it as well.
Yeah, let's take a look.
Just 1 second.
See what's
Is it sonar adjusted at all? Or you have
Yeah, sorry, we're on bottom and trying to get a rock. I'm fine with this dive track. I appreciate the compromise. There's no need to go down quite as deep on the flank below the ridge if folks feel like that's going to eat into time on the ridge. So, but yeah, I do appreciate the compromise. Thank you.
Great. So it sounds like we all have an agreement. Go ahead.
We also appreciate it too, Matt, because it's a compromise both ways.
Thank you, Chris.
Great. So it sounds like we have a solid plan for tomorrow. So I think we can transition now into our dive today on Seamount D. So we're at a depth of 3,404 meters. And we are encountering a nice field of potential loose rocks. So we're going to go ahead and collect one rock right here, and we'll continue with the plan we showed earlier of going over this steep slope and hopefully encountering some nice biological communities along the way.
gonna be, no. That might be too, it's out of my reach anyway. Sorry, I gotta reposition real quick.
This one's a beauty.
So again, today we were diving on the northeastern flank of Seamount D. So this is unknown Seamount. That's previously, before this expedition, was unmapped. We completed partial maps last week, and we finished the map.
Squeeze it really hard.
Seamount
Sure can.
last night. We're diving at a depth of 3,000 meters, and we hope to climb one of the steepest slopes in the Seamount.
Oh, it looks loose.
Hopes to observe some interesting biological communities. And our geologist is very excited about a rock.
That's a keeper.
It's a big one.
Is it too big? If it's too...
I think we can get it. What do you think, Jeff? Yeah, I think we can get that.
That's a beautiful rock.
It'll fit in the box. All right, let's do a quick zoom on this video. Come on in.
When you get a chance, if you guys also maybe deploy the arms.
So that's looking good, Matt?
Yeah, I think so.
All right.
As good as anything can look when it's covered with a bunch of ferromanganese crust.
All right, let's come on out video. Thank you, Jeff.
So this will be specimen 1, the geological specimen in dive 11 on Seamount D. And this American Samoa expedition. We're at a depth of 3,004 meters, and we're stowing the sample in the starboard bio box. The starboard rock box.
That's a little snappy. It's at grip force 9. All right. We can bring that wing in, Jeff.
So the depth range of this dive is going to give us a really interesting comparison to the dive we did on the shallowest portions of Utu Seamount. That was dive 6, and right now we're dive 11 of this expedition. So they both targeted the same depth range of around 3,000 meters.
It's slipping away. It's slipping away. I mean, I'll back up. Just. No, nope. Slopes in just a little bit. All right. We could probably bring out the uppers if you want. Yeah, yeah. Yeah, that's, fully deploy. Break out the light pool a little bit.
So we see some large outcrops coming into view here, and there are some, appear to be some sponges attached to them. There's also possibly a whip coral, so we want to characterize this animals that we see attached to this outcropping boulder. So pilot, can we take a look at the that whip coral on the left?
On the left. Ah, yes, absolutely.
Huh?
I was like, why was I sinking? Oh yeah, I forgot to adjust my Z-Bias. Okay. Come on in, video.
So this is
Partial.
an octocoral weep. Appears to be bamboo coral from the polyps. The shape of the polyps. Yeah, I think I see a couple of the nodes, the dark nodes in the colony.
That's got
Yeah,
like,
this is a bamboo coral.
all right, Roland, come on in. All right, Roland, come on in.
You see this only, all the polyps are pointing in a single plane. They're not pointing in different directions. They're aligned in a single plane along the axis of the skeleton of this coral. So you see from the laser distance that this is not a very large coral. This may be about a foot tall and a very slender skeleton. I didn't see any. Is that full zoom, pilot?
All right, come on in, video.
There we go. Gonna take a closer look at the polyps. Sometimes you see these white needles sticking out of the polyps in these bamboo corals, but I don't see any in this particular one. But you can appreciate the great detail of the polyps, and actually it seems like not all polyps have the same length. They are right now the ones that are running from top down seem longer than the ones that are horizontal, which is really interesting. Normally in octocorals you see all polyps having, or all tentacles in the polyps having the same length.
Let me talk with watch leads real quick.
Video's clear.
Can we get the back polyps?
Correct. Yeah, the plan for the
Can we
whole
zoom in
lab
and
is
get the-
move us
the
slow,
cor-
but
polyps
we'll spend
in the back,
a
Roland?
few minutes here characterizing the animals that we encounter here before starting the move. Thanks. That's great detail. Thank you, pilots.
Yeah, they want to look around here for a little bit and then move upslope.
All right, come on out, video.
That's max zoom.
Come on out, video.
Southish, yeah.
Santiago, while we are looking here at this really, really beautiful
Little east. Yeah.
coral, just wanted to say hi and, well, we are listening from Pennsylvania and Lehigh again.
I think you
Great,
see
thank you. How are you?
160 on the
Matthias.
NS move.
Forward to see.
Let's come in on the sponge.
Thanks for joining us. Should be a very interesting dive today. We're trying to compare the similar depth
Let's come in on the
range
sponge.
between this place at 3,000 meters and what we observe at 3,000 meters as well on the top of Utu Seamount, further north in American Samoa, so more than 100 nautical miles from here, north. So we're seeing here is a sponge.
Max zoom.
I'm not sure I recognize this one. Chris Kelly perhaps can tell us his opinion about this sponge. It's hard to get into focus. It's basically pointing towards the ROV. So you can see it has a long shape coming out to the rock, and I think if we can get in focus the base, I think it has a stalk, yeah. So it has a stalk that is the attachment to the rock, and then the rest of the body comes out above that stalk. The stalk. Maybe we can find another similar one later and take a view laterally.
Ferromanganese crusts here are pervasive on all the rock surfaces, so this is consistent with a really old seamount age.
Yeah, there's several other sponges here to the right and to the left. Actually, seems like there's a high abundance of sponges here. You see this one large in the center. These all look like glass sponges.
Let's get the large one rolling.
Group of Hexactinellida. Just trying to see if there are different kinds. They do, like you see this one has an attachment. You see the base of it is not on a stalk, but it has fibers.
It's a partial.
That look yellow.
15 or 20 I think will be fine.
Compared to the more...
Yeah.
The other ones on the sides of it that are raised on a stalk. They're not attached by these long filaments.
20 meters, 160.
So I think we have a couple different...
Yeah, sorry.
Go ahead, Chris.
Yeah, sorry. I ran up to my office for a moment during the rocket lecture. I didn't think you'd get into biology, and you sure did.
Yeah.
The bigger sponge is one of these sphaeronematids with the lophophytus attachment. That is that fibrous attachment, and it's probably, again, in the genus Polyopogon, because this whole family is very poorly described, so we kind of default for a lot of the sponges to that genus. But it looks like what we've been calling Polyopogon. This one that's to the left with the stalk on it, this is not a glass sponge. So the big one you're just focusing on is a glass sponge. This one is actually a demasponge, and it's something that we're currently calling Stellodorix, and it's in family Vulcanellidae, and this is coming from Dr. Rob van Soest in the Netherlands, who we consult on this group of sponges along with Shirley Pomponi as well. But it's very unusual. If we hadn't actually collected some of these things and took a look at the microscopic parts, the spicules, I would have automatically considered this a glass sponge. So there's lots of surprises down here in deep water.
Absolutely. I definitely, when you see similar morphology like this and the colors, and I couldn't really tell it apart from a glass sponge at all. So we see several of those small individuals of that demon sponge that has a stalk. What can you remind us of the genus or species identification, Chris?
So the genus, and I'll go ahead, I hadn't gotten into C-scribe either. I was getting to the ECC a little late this morning, but the genus is called Stellodorix, and it's in the family Vulcanellidae, and of course that's all a guess without a specimen in hand. That's our best guess based on its similarity to other specimens that have been collected.
Right.
I also wanted to make a remark. When I was up in my office, I quickly put on the video. I'm actually downloading files from the FTP site, and I saw that you had an unbranched coral there, and I don't know if anyone ID'd it, but that looks like Heratocipes to me, Chrysogorgid. Is that what other people thought, or?
The one we just imaged, Chris?
What's that?
Are you referring to the one we just imaged?
Yeah, as soon as I saw that, I quickly got my computer going and copying things, and then I ran back down to the ECC, and you were on the sponge already.
Oh, that was a bamboo coral.
Unbranched sort of whip coral?
Yeah, it was an unbranched whip coral.
Did anyone come up with an identification on that?
Partial.
It was a bamboo coral, but not as far as the genus or species identification. But it was a bamboo coral. It wasn't a Chrysogorgid.
Okay, Okay, yeah, I didn't get a really good look at it. I had it on a small screen, and at first I thought it might be one of these unbranched chrysogorgids that are quite rare, but I got
Okay,
all
but keep
excited.
an eye out.
Right.
Should have
Let
looked
me
at it
know
a
when you
little
think
more
you start to
closely.
swing.
Right, thanks.
So keep
Thanks,
track.
yeah. And what we're looking at right now is this is chrysogorgid, the ones we observed at the similar depths, what we've been calling a Plurogorgia octocoral. And right now you see most of the polyps has the tentacles retracted. And this one has a black skeleton. We observed this also at the similar depths and the
you've moved a little bit.
northern portion of the northern seamounts that we explored
So that's only
last week.
3-minute delay.
Yep, go ahead.
Is that an Opheroid, a transparent Opheroid?
Let's come in tight.
I see a shrimp, a mysid shrimp. I'm not sure if that's what you're referring to. Oh, yeah, there it is.
Oh, good
More
eye.
down.
Yeah,
It's a translucent almost.
yeah.
This is a beautiful. Wow.
Yeah, we've seen several of these. They do resemble Opheroids, but we've never seen one move so far. That's
If you sampled this coral, you could get that as a specimen if it held on.
yeah. Yeah. Wonder if it's
Ready for a full?
Yeah, come on
interest
out.
in Fox to do people on shore to be able to compare these corals to the ones we saw further north. They do resemble the same morphology.
Something growing.
Couldn't
in
say if they're the
coral
same
to right.
species, but might provide
Probably an enemy.
information about the range of, if it is in fact the same species, of how far south, because this is the furthest south we've seen it so far.
All right, come on in.
And
Roland.
this is actually the furthest south we're going to get in this expedition. So you
Starfish.
see this
Anemone.
anemone.
Wow.
And this actually looks like a different kind of coral. So we can get a closer zoom on the polyps. Yeah, so this is a different kind of coral. This is a primnoid coral. You can tell by the coloring of the skeleton. Looks a bit more golden. But also, if you look at the polyps in detail, I don't know if we have full zoom. I'll be able to
Do we have
see
any more zoom?
better what I'm
Smack zoom.
talking about. That's max zoom, okay.
I can get closer.
But you can you can see that the polyps have these plates
Partial.
that are like armor polyps that are characteristic of the family Primnoidae, these octocorals. So right now we've already encountered representatives of 3 different families of octocorals. So the bamboo corals, the family Cetidae.
yeah,
The
back off.
coral we just imaged previously had a dark skeleton, but they're commonly referred to as
White.
golden corals because most of the species have a golden
Nice
coloration
spot,
in the
yeah.
axis, in the
I
skeleton.
didn't see that overhang.
And this is a primnoid octocoral.
Ooh.
And these 3 families are the most commonly found at these depths in deep sea worldwide.
Yeah, come on in,
Any
Roland.
chance we could look at that stock sponge if we're headed that way after the primnoid, after you get a good image of that, and the anemone?
Okay, come in,
Sure.
Max.
We can, after we image this primnoid coral, we can take a look at the stock sponge. Are you referring to a particular stock sponge, Chris, or just in general, the ones we've been seeing?
No, there was one in the upper left when he was zoomed out a little bit more, but let's talk about the primnoid first. I think it's Norella, but I don't know what you're thinking, Santiago. A couple of the polyps are closed, and they close down towards the base of the colony, which is a characteristic of Norella. The anemone, I think, could be an Ectinoscyphia, another one of these Venus flytrap anemones that you see on corals and other types, and sponges sometimes.
Yeah, this is another of those Venus flytraps. We had a really nice behavior display a couple of dives ago. Yeah, we observed these similar kinds of primnoids in the dive Seamount at the same depth range. There we were seeing different sizes of these primnoids. I'm not sure if this is exactly the same species, but we saw several that were just whip
yes.
morphology, but then when we saw larger individuals, they had branches, long branches like this one. So basically like a
that's like 4
V
minutes
shape with not a lot of branching, but long branches when they were branching.
Are you good with this watch sleep?
Yeah, thank you.
All right, come on out, Roland.
So that actually, I was wondering, we didn't collect that particular coral last time, so wondering if it will be an interesting collection, because I don't recognize this coral really, and it will be also interesting to sample that associate, the Venus flytrap that we've seen also, but we also haven't sampled, characterized what species it could be.
Yeah.
So it might be a good opportunity to constrain the species that we find at the depths of around 3,000 meters in the American Samoa region. So what do you guys think in Hawaii and maybe with Saul and Tara here in Stockton?
Well, I'd support it. I mean, you're also going to make somebody in Smithsonian extremely happy. That's Steve Cairns, who works on primnoids.
Yeah,
and,
that's true.
Yeah, I think it would be very useful, and the fact that you get the anemone as a bonus is a good, point.
Right, we'll be able to characterize the associate this coral. So, Pilot, we are. Go ahead. I think that's Tim.
No, Tim, I agree with that. I would love to
learn
learn
a
more
little
about
bit,
that
Jeff.
anemone, if it's an Acumis gysa or something else. It would be a great opportunity
copy that.
to get that pairing of that coral and an anemone. So I'm in support of a sample.
Great. I think that's good to hear the feedback, and so we're gonna try to get settled here and sample this coral. That
come out a little on Sirius.
From my observations in my notes, it does seem to be very much resembling the one we observed further north to Seamount at the same depth range. But at that time, we didn't have the chance to sample.
Yeah.
So this will be a really good value sample to be able to identify the species.
I can get it from here.
And also the identity of that Venus flytrap anemone.
The anemone? Yeah, the Venus flytrap. Yeah, copy that. A little hot on Zeus video.
Yeah, it looks like we have an overhang here near the ROV. If you look at the Sirius view.
Can we cool
if
down
I'm viewing that correctly?
Mini Zeus?
Pretty spectacular overhang,
Yeah,
actually.
I agree, Matt. I see it. It's a little bit of an overhang, a little rock jutting out there.
Yeah.
It's hard to tell if it's like a lava pillar that's above it.
Can you look
off
with my utility cam, Jeff, and just make sure my arm's not going to hit this rock? Yeah, okay.
And
come in a little on Zeus.
Okay, and saying what?
All right, hold that.
Oh, okay. Let me know when. Yeah. So RV nav. RV nav.
Oh.
Yeah, I'm going to have to move to get the box open, I think. Yeah. All right, you can try it. Well, we can zoom while we're doing this. You can go ahead and zoom, Roland.
Okay.
That's a much greater detail of the polyps. And you can see this polyps have basically an armor made of sclerites, of calcium carbonate sclerites.
You're hitting that coral. Just hold there.
You can see some
come on
of
out.
those sclerites have spine projections coming out of them. Those are important characteristics.
Tilt down
And
on
also,
the just a little bit.
as Chris was mentioning, the disposition of the polyps, where they're looking up or down relative to the position of the skeleton. So this is the second specimen that we're collecting today. This is a biological specimen. This is a primnoid octocoral with a Venus flytrap anemone associate, and it's going into the inboard port biobox. It's a great sample. It's now
clear.
Fell into the biobox, and now we are sure that it was a successful sample.
By the way, I just wanted to point out that one view of the close-up of the branch is essentially a microscopic view of the coral polyps. That's just remarkable when you think about it. I mean, that's literally what we see when we're looking at a microscope.
Yeah, that's fine.
Yeah, I agree, Chris. It's amazing the level of detail we can get with these cameras and the lighting system, and obviously the skill of the pilots and the engineers to use these tools.
Do we come wide on HD2 on Sirius cam?
All cameras are full wide.
Ooh. Come up on.
I'm sorry, Sirius is not. Sorry.
Yeah.
Sorry, gentlemen.
Yeah, go ahead. Oh, the ones right in front of us? Yeah, we can zoom on them.
Chris, is that the stock sponge that you wanted to image? I want to take a closer look.
Yeah, go ahead in video.
Yep, that's the one. I don't recognize the sponge. It's got a large osculum on top of it, and then it's got these droopy sort of protuberances coming down from the main body
ahead
of
in
it
video.
toward the
Don't they want to see the stalk on the very top?
They want to
left?
see the rocks.
Oh.
Great, so I think this is a good spot because we can check those rocks out and see if they're collectible, and also it's interesting imaging
come on out a little bit.
Just
sponge
getting the
afterwards.
Yep.
Partial.
Let's come out just a little bit more.
Partial.
Oh, that big rock is beautiful, but it's too big.
Too big, unfortunately. There's a little anemone back
Wow.
there, too.
That's a big anemone, yeah. That'd be great to image that, and
I'd
as
take
well
that
as the
big
sponge on the
rock
left.
with the big anemone, and we'd all be happy.
Come on in video.
Had to leave the other rocks behind.
No, say it isn't so. Say it isn't so. I'm really impressed with this overhang. Trying to get a better sense of it here.
Wow, this is a really...
That's a great view. Look at that. It's just framed in the between the rocks there. What a nice shot. Beautiful creature.
Wonder if this is a real anemone or...
No, I think it's
there's
plastic.
Well, no, there's
Maxim.
another group of Nigerians that was
Trying to get
recently
my
described
toes
to
in here.
be
There we go.
separate lineage from the, the common anemones that we refer to.
And that last
No, there's a little tag there that says made in China. You see that off to the left?
Mm-hmm.
Yeah, Santiago, that's the Relicanthus daphneae, is that one that was originally considered to be an enemy, and then they moved it out into its own group of Cnidarian. But the tentacles on that one are usually a lot longer than this. I don't know what we're looking at here. It must, maybe even I don't know, it could even be a hydrozoan for all I know. I have no idea.
Yeah, I also just thinking about the length of the tentacles, but this one just looks so unusual to me. That's why I was wondering where.
Come out just a little bit, Roland. Yeah, right there.
Do that, it makes the image a little hot with the rock.
All right, you can come in tight again.
Max zoom.
All right, when you're good, video.
Video's good, we should laser it.
Yep.
Okay.
All right, let's come out.
Great, and I think we want to take a look at the sponge on the left right here.
Yeah, copy.
And there's another coral that we want to see in the background. So they landed in a really good spot for both rocks, I think, and corals.
Nice job, Nav.
You see any rocks here, Matt, that may be of interest?
Thank you.
Go ahead, video.
The rock to the right of the sponge, or maybe even the one the sponge is on. They may be too big.
Yeah. The one to the right of this might be good. I gotta take a look at it with the lasers.
Yeah, so what is this guy? It's a glass sponge. I got that far. I think it could be a stalked euplectellid.
Fill the screen.
But what it is, it could be something somewhat related to the Saccocalyx, which is also a stalked euplectellid that has protrusions coming out of the walls of the sponge, but this just has a few very large protrusions, not like Saccocalyx, which has bumps all over it. So it's really quite an interesting sponge. I wonder if they could pan down the stalk a little bit at some point.
There you go.
Just to see the attachment. Yeah, so that's going to rule out Hyalonema and a whole bunch of other sponges as well, just because that's what's called a basiphytous attachment, where the sponge actually glues the stalk to a rock with silica, versus the fibrous attachment that we saw on that other big sponge a little while ago.
Go back up to the attachment.
So again, best
sponge
guess is some type of a stalked euplectellid in the subfamily Bolosomini.
Thank you, Chris.
Go for a full specimen.
Really nice
Right,
image here.
out.
You can see both parts of the side and the top of the anemone.
That's too big.
Is it? Ah,
Yeah.
say it isn't so.
That's 30, 40 centimeters
Oh, yeah.
long. I don't think that'll fit in our box.
You need something about half that size.
We're not going to be setting the record today for biggest rock.
And yeah, that coral that is on the left, there's actually two corals right now. That's slender.
I mean, we can get it, but
want to take a look at both of these corals.
Roger that.
The whip one might be the same, maybe a bamboo coral, a primnoid coral, like the one we just collected, and the one in the back, it's a different kind. That I don't think we have seen so far.
Right, go ahead and roll.
Oh yeah, so this is what I've been calling just basically the earlier stages of that branching prenoid that we just collected. As just maybe we'll get an opinion whether anybody else thinks it's something different, but just by the structure of the polyps and the disposition, and also just by the observations we made last dive, there were different stages of some that were just whips and they got a little bigger. They had one branch and then some more branches. To me, this resembles to be the same species, just at different stages of development.
Yeah, no idea, Santiago. It certainly could be. It's unbranched, and the polyp structure looks fairly similar to that other one. I think I saw a spine on one of the body scales as well, which is similar. So that may be absolutely correct.
Yeah. And I don't see really any associates on this one. And we can take just a little bit further down. We can take a look at the attachment. Oh, actually, you see a branch there that broke off.
Oh,
right at the base. So it is a whip. Just because it got broken off. Yeah. So there's branching. So this is a good, also piece of information to understand the morphology of this coral. And
right.
I'll see we get into focus. Yeah, looks like a tube on the ground, and Roland was just video engineer. Roland was just pointing that out. That looks like a tube of, yeah, could be an anemone, but there's nothing sticking out of it right now. This is great. So I think maybe now we can take an image of that other coral that is in the background right there.
Let's get this guy in the background. Go ahead.
Now we'll get enough lights on it. Seems like just at the, oh yeah.
Wow. I'd love to have that image hanging on my wall. That's beautiful.
Hey, that is the nice dark background.
Yeah.
So.
Well, my guess is Chrysogorgia. What do you think, Santiago?
Yeah, I was gonna go with Chrysogorgia based on the shape of the polyps, the look of the polyps, and I don't see any banding that may suggest otherwise. It will be great to take a look, maybe see if there's any associates on it near the top. I think that's probably close to the top of the coral. I'm not sure what that is.
That is.
Oh yeah, there's some tiny thing there. Maybe not amphipod, but I think it's out of reach from the camera. And then it would be great also to take a look at the base of these colonies to see what the color of the underlying skeleton is. If it reveals any. Huh. So it looks like it has living tissue covering the whole skeleton. So I'm not gonna get a good view of its color underneath. So yeah, I think it is a Chrysogorgid. I don't know if it's a genus Chrysogorgia. It doesn't have the bushy morphology that's characteristic, but I mean, it might be a juvenile or maybe another completely different genus.
Yeah, come on out, Roland.
Almost looks like X-ray.
I'll be interested in getting the rockets attached to. I know it's an old joke, sorry.
All right, anything else here?
No, I think that's good, thank you. We can continue exploring these outcrops. Seem to be encountering a good diversity of species here. Oh wow, look at this sponge, maybe we can take a look at it.
So my best guess on this one is just a version of the genus Phytoplura. It's a hard, solid sponge, tend to form these upright colonies that can be very paddle-shaped. But then again, it's got a lot of channelization here. Those are those grooves down through it. So best guess is Phytoplura at the moment.
Excellent.
All right, come on out a little bit.
Can you provide folks with a sense for how fragile this is, Chris? If it's collected, how...
Come out just a little bit more, Roland.
Well, those ridges and then the areas, the raised areas between the ridges themselves are going to provide it a certain amount of durability, so it would just snap off or break. It wouldn't just crumble into a whole lot of pieces like a lot of sponges that are brittle sponges. But we're seeing so many weird animals that, including this one, that I don't know, seems a bit early to grab a second biologic collection. I think others probably would agree to.
Yeah.
Let's see what else we find first before we start making, before we do another collection.
I'd say this is for me as a geologist, this is the most beautiful sponge I've seen.
Come on up.
It's like it's something out of
lasers.
Full screen.
Out of a Giorgio O'Keeffe painting.
It's about 60 to 70 centimeters tall.
If you actually dissolved away the tissue on something like this and looked at it under the microscope to see the microscopic skeletal parts of the sponge that are called spicules, you would be absolutely amazed. There's just an incredible array of different sizes and shapes and beautiful spicules looking like snowflakes. And that's, all of these colonies can be really gorgeous. The architecture of them is equally gorgeous when you look at them under a microscope.
So at all length scales, you see beauty in these sponges. Oh, here's a nice outcrop. So this is the overhanging outcrop we were looking at before.
Yeah, this is an Iridogorgia. Corals are different species of Chrysogorgid. The spiral.
Come on in, Roland.
Morphology of the skeleton is characteristic. There's actually two genera that are closely related. We did again observe this kind of coral at the similar depth on U2. So far, most of the animals we've seen in this dive, we also saw at similar depth on U2 Seamount. Perhaps a couple of the sponges I don't recognize. You see the nice detail. The octocoral polyps are so distinctive. And maybe as we zoom out, we can take a look at whether or not this coral has associates with it.
Come on out, partial.
At U2, we observed that one of these kinds of corals was being predated by a sea star.
Partial.
And also we've seen these corals at shallower depths with squat lobsters associated with them here in American Samoa. See something, one of the branches are right above the lasers pilot. Yeah, that right there.
Extended.
That looks like a ctenophore. Yeah, that's a ctenophore that is attached, one of those benthic ctenophores attached to the branch of the octocoral. You can see the tentacles projecting out with small lateral projections from it, and using those sending into the water column to capture particles. So those have sticky cells and they can retract them to the center of the body and bring the food particles to their mouth.
So sticky, but not poisonous.
Yeah, that's right. So the jellyfish.
This is a really unusual bentactinophore. You see that it's branched, the pinnae are branched, and see those filaments coming off laterally.
Right.
So that is not like the Tijalfella that's in the OER animal guide right now. This is probably something more related to the Lyroctes, another type of bentactinophore. So this is probably the first time we've seen this animal, and it's just astonishing the variety of different animals we're seeing at the site. It's really incredible.
Yeah, I agree.
The tentacles were exceptionally long. I had no idea.
All right, come back in.
Yeah, we saw. Remember what you call the disco ball, the red one?
Yeah.
But that was a pelagic ctenophore.
Oh,
so
okay.
It's essentially the same group.
Oh, I see.
But this one is attached to the coral.
The tentacle looks to be 100 times longer than the main body of the organism. At least.
Yeah, so you can get, it's a good strategy to create these structures that you can extend, just cast the net essentially into the water column.
What's impressive to me is those tentacles don't get tangled up either with each other or with the associated coral.
That's true. They might be, evolved in a way that they are able to capture particles, so be sticky to other particles, but not to themselves. Or maybe it's a behavioral component to it that prevents them from sticking to themselves.
So how is it attached?
That I don't know. Seems I don't know if the tissue goes all around the branch or whether it's just cemented or a glue that it uses to attach itself to the branch.
Were you saying before that these are their own phylum?
Ctenophores, yeah, they are
impressive.
Phylum. They are different from the cnidarians, like jellyfishes. Like this coral right here, where it's attached, is a cnidarian. So are jellyfishes, which when they are the jellyfishes in the water column as well as ctenophores, they're, they can look pretty similar. Just because they're gelatinous organisms, but they're quite distinct. They're unique. Thank you, Pilot. This is a great image. Is that full zoom, I imagine?
That's full zoom. I could probably get a little closer.
No, I think it's fine. Thank you.
All right, copy that. All right. Let's leave that rolling.
We see an abundance of smaller sponges on the right edge of this outcrop. And we see them a lot more also on the next outcrop on the right.
Base, these are all pretty big. These are the ones we were looking at before. Yeah. Come in partial video. I mean, the thing is, I can't get in too far in there either because of the overhang. Yeah, we might find a better spot moving forward. All right, come on out, Roland. Okay, copy.
So as we rise above this large boulder, seems like these are detached boulders that just roll down.
Yeah, I'm thinking something similar, yeah.
Let me know if there's anything you want to zoom in on.
Yeah, maybe we can take this couple of corals that are coming into view.
Yeah, copy that.
Maybe this one in the center.
Candelabra style.
Maybe just do a snap zoom if you can. They look like the Plurogorgia.
Video.
It would just image. Just want to make sure that's what they are. Yeah, this does have the morphology of that coral, the Chrysogorgia plurogorgia, like specimen or species that we've been seeing.
Come in tight.
That's one of the most common deep coral fauna on these depths. Yeah, this is good. Thank you. I think we can.
Come on out, video.
And there are some very small sponges to the right, and maybe we can take a quick peek at. Thinking maybe that one that looks branched. I don't think we have imaged that in detail. The city one in the center.
Coming in.
Oh, a big boulder in front of us balancing precariously.
Okay, come on in, Roland.
Matt, I think it's too big to collect.
Oh no, don't say that. Don't say that. Let's be positive.
Yeah. So this looks like a sponge to me too. Chris, do you recognize this?
Maxim.
Well, I think it's a demasponge. Again, these Stellodorics, or what we're calling Stellodorics, seem to have a variety of morphologies. We seem to have found some that look almost like planar bushes. We also have ones that, as we imaged before, that kind of look like a sock on the top of a stick. The only thing we can do right now is just go to demasponge, possibly Stellodorics, for all of these that are hanging out on the ledge. So that one too, over to the left center, we're calling that Stellodorics as well. At some point in the future, we'll probably quit defaulting to that when we know something better about them.
Great, thank you. Yeah, I wouldn't know. I guess this is a demasponge.
Yeah, come out video.
All right. Thanks, pilots. Maybe we can start moving ahead.
Yeah, copy that.
So we've been on bottom for about 1 hour, and we've moved maybe 20 meters. I don't know if there's interest in moving along here.
I think the answer is
I
yes.
see some possible rocks for you, Matt.
Unless, of course, we see some rocks.
Yeah, but that was a great spot to start the dive on because it had a great diversity. So we able to one spot to characterize
I don't know.
Several different species that we will likely continue to encounter along this dive. So for those of you that missed the earlier description of where we're diving at 3,000 meters on the northwestern slope of Seamount D. So this is on previously unmapped Seamount. Doesn't have an official name. We've just been calling it Seamount D.
Any of these pique your interest?
And we picked that dive side that was basically we're between two ridges. And
lower left.
Do you think we should
we
put in a
are
short move?
on the
lower left. We're going over it.
We do 15.
Backing up.
If you look at, if you were imagine looking at a ridge, looking from the top, we're diving. So we're looking at the ridge and going up slow. We're diving on the right side of the ridge. It's one of the steepest areas in the Seamount, and that's why we chose it. And
that guy?
Also by being between these two ridges, it's a really interesting area. We thought we a good habitat where we have provide
this one
vertical
is fine.
walls.
The one the lasers are on
very scarped
is
terrain, like the one we're seeing now, but also funnel potential currents
15,
that might provide an enhanced food supply and greater abundance of biological communities like sponges and corals, like the ones we just observed.
175.
Yep, that one's loose.
How about the one over to the right a little bit? It's more angular, just to the right and down. To the right and that one.
Oh, the small one?
Yeah, it's smaller, but
yeah, we can definitely
looks
get that
less
guy.
likely to be a manganese encrusted ball of mud, which is always the concern here.
All right, guys, we have a short move, 15 meters at 175 degrees initiated.
Copy. Hydraulics up. Alright. I'll give this guy a squeeze. Grip force 9. So that's full squeeze.
Full squeeze, and it's not breaking.
It's not breaking.
Oh, that's nice.
That's nice.
Yeah, that doesn't look like mud.
Alright, I'm going to just set it down here.
So is that 800
It's
pounds of
small,
force?
but some of the biggest ones I've obtained have been... Yeah, that looks like a chunk of a pillow. That's a nice rock. It's not the kind of image I'd want hanging on my wall, but it's still a nice rock.
Can we reposition HD2 for the collection?
What? Why do we need HD2 for the collection?
In Stream 3, I do a quad split of those cameras and the two manips for them to see back on shore. And it shows them which rock box it goes into visually.
Yeah. Just stand by it. Just gotta get set up here. Alright, good.
Right, so
Tilt
this
up my
is the
juice.
third specimen
Get it, Jay.
of this dive, and it's a geological specimen, and it's going to be stored in the port rock
Oh, no.
box.
Ah,
But the specimen just roll away.
tilt down.
Still there, somewhere.
Where'd that guy go?
Oh, this could be quicksand.
Is he still in there?
I think it rolled under to the right. The dust continued to the bottom right.
Look at him. You bring that, well, leave it for right now.
I don't see it. I think it might have rolled downhill.
No.
Where did he go?
I think it may be coming.
That's not it.
I think it may just be above that rock.
Couldn't have gotten that far away. Isn't that one?
Isn't it,
Oh yeah, you can see the track
am I
there.
mistaken, or is it not further up?
Oh, is that it?
Yeah, you
Yeah.
can see the track
Is that
of
it
the
right there?
sliding right there.
We gotta bring
yeah.
that wing in. Unfortunately.
It's a good thing that a rolling rock gathers no sediment, because we wouldn't see it otherwise.
Almost started a landslide
don't
here,
push
man.
Stupid joke,
Rolling,
wasn't it?
rolling rock. Rock roll.
Yeah, that's good.
Rock and roll.
Rock and roll on the sea floor.
You tried to get away, watch lead,
Isn't there a
Oh, we
rolling
found him
rock
again.
beer, too?
Rolling rock beer and rock and roll. That's
yeah, I know. Yep.
I've made no beers.
Yeah, I see that. Yep.
Video swapping out.
Can I even get to that? Yeah. Could bring that wing all the way back out. Outboard. Yeah. Thanks. Watch my starboard. Yeah. Think that's out of reach? All right, let's do it. No, I'm gonna put it in the. I'm gonna put it in the port. All right, go ahead. Man. Yeah, I'm not super used to grip force 9. Closed a little quick on it, which is why I lost it. It's like all or nothing, basically. Yeah. Good. All right. Took a little longer than normal, but we got it, Matt.
Josh, thank you. ROV team, thanks for making this happen. That was a great sampling effort.
It's our pleasure.
It's a great sample. And, wow. It's a great sample and gave us an example of a rolling rock on the bottom of the sea floor. 4 kilometers.
Hey, Pat, I missed that. Did that go?
That's ROV rock and roll. That rock rolled.
Got it.
I guess 4 meters or so from where it was first picked up. So it gives a sense of the steep slope that that we're looking at right now.
So Sirius should be just
you can bring
on
out
the
that
swing.
starboard swing arm again.
Right now we're having a change in pilots. We're at a depth of 2,984 meters. And for those of you may be wondering whether we move really fast from 4,000 meters to this depth, we actually had a last-minute change in the dive plan as we reviewed the maps this morning.
Since where ship stopped.
This was on map, previously on map Seamount.
Okay.
So we actually didn't know what the summit or the shallowest portion of the Seamount were.
2 rock samples, one
and
in
given
each
time
box,
constraints,
and
we decided to
1 biosample in your port inner.
do a shallower dive to 3,000 meters.
Sorry, can you repeat?
That way we have a
2
longer
rock samples,
time in the bottom.
1 in each rock box.
We still have
and
around
then you have a biosample in your port inner.
3 hours left of bottom time.
Yeah,
So how long was the
game
descent?
plan
How
is
long
pretty
did it
much
take
just
the
mid-up
ROV
slope.
to descend to
We have no hard
3,000 meters
targets
on
at
Tiago?
descent. Waypoint 2 is pretty soft.
That was 1 hour and 40 minutes.
Okay.
We've only
So
made
the deeper the dive, the longer it
35
takes for
meters
the
of ship
ROV to
distance
get to the
accumulated
bottom.
Oh,
in
really?
an hour.
Oh.
That's right.
A lot
And
of samples and zooms. And
descent
a lot of
rate
stuff to
is
look at,
about
so I think,
30
Washington's
meters
one
per
of the
minute,
little
so that's about
quicker.
90 feet per minute.
Just gonna get the Z-bias dialed in here. It's floating upward.
It takes just as long for the ROV to then return to the surface. So we lose, the deeper we dive, the less time we have each day
And the ship, Sean says
on the
the ship
bottom
is
to look
stopped and
around.
probably
yeah.
settled out by now. But just want to get the Z-bias adjusted.
Last move was 175, but it's been adjusting plus or minus 5-10 degrees. So whatever you focus on an upslope, that'll work.
So what are these trails here, Santiago, in the mud?
They appear to be
Yeah,
trails.
Sirius hasn't had very strong returns.
Potential bioturbation trails.
Okay, I'm going to come back
You
down
see
to the bottom.
two different things. The ones that are running down slopes, those appear to be ripples created by currents.
Those are the settings Josh
Suggesting
told me
an left to right or right to left current direction.
Correct. So the
right.
current heading is...
So make Sirius...
Oh, watch lead, this is pilot.
South. Go ahead, pilot.
Yeah, we just had a shift change. Oh, sorry, let's stand by. They've got ship alarms going right now. Yeah, we just had a shift change. I'm Dan Rogers in the pilot chair. We've got Andy O'Brien as co-pilot. And Sean is still in the navigator seat. He'll be replaced by Carl McCletchy shortly. We have Bob Knott in the video chair and Emily Narrow selecting our highlight clips in the back. And we're all with the Global Foundation for Ocean Exploration.
Yeah, it's great to have you.
Okay, I'm kind of come down, Andy.
Absolutely excellent ROV team and team of engineers we have on board. We actually haven't done introductions today, so the people here in the back row. So I am Santiago Herrera. I'm a visiting assistant professor at Lehigh University, and I'm the scientific lead, and I'm a biologist on board, representing also the interests of the broader scientific community interested in biological specimens that we observe during these dives. And to my left, I have
Matt Jackson. I'm the geology lead here on this ship, and representing the geology interests for this expedition. And I hail from UC Santa Barbara. And I have been trying to get to this seamount and a couple of other seamounts in this area for years. And we tried to dive on this seamount last week this time, and we had to abandon the dive. And the ROV was already a few hundred meters depth.
Do we want to look at this creature here?
Yeah, can we do a snap zoom? I think we might have an idea what it is. So
yeah, let's go ahead and zoom in partial video.
Tropical storm Bart chased us away.
There.
So we're now, this is our second attempt on this seamount, and we're really excited to see the rocks
here.
here.
I already introduced you on comms.
So this looks like the
Go ahead and zoom in.
Bamboo coral I seeded that we observed image earlier.
Hold there. You imaged this one already?
Yeah, we took a good look at it just earlier, so we just wanted to confirm the identification from the distance. Thank you, pilot.
Okay. All right, Nav, Sean was saying that the ship has stopped. Sirius has probably settled out, and we're heading at approximately 170, I think is what he said. And he just said we're heading upslope, and there's no hard waypoint, so.
Yeah, we're still kind of in the flat right at the bottom. It should be transitioning to steep.
Okay.
This is some of the most extensive sediment cover
pretty
that
well
we've
ahead.
seen on any of the slopes of the seamounts. And
see, if we do a 30 meter and they want to stop right away.
Maybe 20 meters.
Okay.
We can do that.
Yeah, so if you start going ahead now, it should come down.
Is that a Holothurian up top? The top of the screen? Well,
this looks like another whip coral.
At the top.
On the right.
Yeah.
Let's take a look at it.
It might be a Holothurian. We saw the other day with the sail. Go ahead and zoom in partial.
Oh yeah, it is.
Hold there.
Good eye. The other one we saw was purple and larger. This is only about 10 centimeters long. Actually, the sail, the projections coming out of it are longer than the length of the body.
Yeah, come out a bit.
So this is something that we hadn't observed. We haven't observed so far in this dive or in this expedition. You see that it's the sail, so that there's like seems to be have three projections or three lobes.
Co-pilot, according to HIPAC, it's more like 140.
We observed
just
one.
Follow the local,
it was purple and it had two lobes only.
The goal is just to go from the bottom to the top, kind of perpendicular.
So Santiago, this is most likely in the family Elphidiidae with that huge sail and the very clear body and those 2 feet on there, and it may even be something that's similar in the Okeanos guide, Amperima species. Those IDs are coming from a couple different folks that specialize in this in both Russia and at the Smithsonian. So anyway, that's the best guess on this one. I'll punch it into C-Scribe.
Thank you, Chris.
You can see what it's eating because the body is translucent.
Yeah, right now we can see it. We can watch it eat. You see it's extended some of those feeding appendages. If you tilt down the camera, Violet,
sure.
there you go. It's going and grabbing pieces of sediment.
So 170 at 20 meters.
And likely bringing it into their mouth.
175.
The sediment's coarse, light colored, a lot like the sediment we've seen elsewhere.
Yeah, resembles a lot of foraminifera tests.
Copy that, Bridge.
Likely calcareous.
Darker bits as well that might be pieces of basalt that have broken off, or ferromanganese material that's broken off from above and rolled downhill.
So is the sail used for swimming?
As far as I know, it's
zoom out a bit?
unknown.
Unknown?
Yeah, they do swim, but it's not clear that this is...
There's a little something at the top. You want to zoom in?
An active swimming structure. It might be
little
just exactly as a sail, maybe, to get a better
co-pilot, let me know when you start moving.
Area and traction with the currents.
And, get an
I'm
idea
just
for the
speculating
delay.
here, but
okay,
it's
let's go
possible.
and zoom out.
You can tell, yeah, the sail is, as I said, looks like it's longer than the length of the full body of the animal, which is amazing.
This is about 10 centimeters wide, Santiago.
Yeah.
That little creature is 4 inches wide. 4 inches long, rather.
Okay. All right. Let's go see what we find next. So we're going to be transitioning into some steeper terrain, co-pilot, so if you could help me keep an eye on my swing arms. At some point I'll just have you bring them in, more than likely.
Okay, the ship started moving 5 minutes ago. I'm not sure if Sirius has felt it yet.
Okay, I'll just...
Okay. Yes, you can scan side to side.
Yep. See what kind of slope we're on now.
So I see a couple more corals and perhaps sponges on the right. It's maybe a different kind of coral. Let me take a closer look.
Okay.
Sure.
Sounds about right.
Go ahead and zoom in,
5
partial.
minutes.
And hold there.
Yeah, don't show this to Les.
Yeah, this looks like that slender, partially branching chrysogorgid. We saw this also on Seamount. And in the background, that looks like the whip is seated bamboo coral. See the internodes in the back, but yeah, the one in front, it looks to me very much like a chrysogorgid octocoral. We just also observed one of these colonies just a few meters deeper as we landed on the seafloor. I don't see any associated. I don't know. There's an amphipod clinging to one of the polyps. If you tilt the camera down, there it is. So actually, that amphipod may be not directly on the polyp, but on one of those what we've been calling clear opheroids, the figure 8 opheroids.
Okay, let's zoom out.
Interesting. All right.
See a stalk up ahead.
That's on the right. There's the whip bamboo
Yeah, this looks like a debris field here. Large blocks, boulders that don't appear to be attached to the substrate on a relatively steep sediment covered
One single
slope.
stocked sponge.
Santiago, Tim, did you say you thought there were Opheroids on that Isogorgia?
Well, Tim, we've been observing this clear animal that resembles in morphology to Opheroid.
Hold there.
Very small. We've seen it on sponges. We've seen
Definitely
it on corals.
a starboard to port current today.
We've seen it on hydroids. Is not moving. I haven't seen it move. And we actually did collect one specimen of those with a hydroid. I still haven't had time to look at it on the microscope yet. Still been on my list of things to do. But there was amphipod on top of that, what seemed to be a Opheroid, yeah. Which was...
Okay, thank you. Yeah, I thought it looked like a hydroid when I first saw it.
Right.
Okay, go ahead and zoom out.
But then you said Opheroid, and I wasn't sure.
Yes.
Okay. So we're looking at a Colophacus glass sponge. That's a genus in the family Roselidae. It's one of the, it's actually a stock sponge, but it's different than that other stock sponge that we saw, which is in a different family. Of course, anyway, this one also has beautiful microscopic parts. It's very common in the deep sea, and we seem to see that particular genus all over the place. But it's always a great one to see. We used to call them pom-pom sponges many years ago before we understood what they were.
The sediment cover here is really extensive.
Yes, definitely an area where there's a lot of material rolling down.
Really, some of these blocks have got to be 10 meters wide, and they're not. They don't appear to be attached to the substrate.
Big ledge there. Coming back down.
Can imagine there must be some potentially large landslides here whenever there's an earthquake in the area.
Ooh, creature over there, another Holothurian maybe. Go ahead and do a snap zoom over there.
Oh yeah, good eye. It looks like another.
Oh, it's a
that's
fish.
a fish.
On the sediment, or is that
a halosaurus.
Yeah, it's a fish.
Looks like a halosaur fish. We don't have Bruce today with us, I don't think.
We can do a quick tighter zoom.
Yeah, so you're absolutely right, Santiago. It is a halosaur in family Halosauridae. If they could zoom in on the snout, that's the key for diagnosing what genus it belongs in.
Go ahead and zoom in. Oh, he's gone.
And so it looks like it does not have a scaled nose, if you will, that area between the eyes and the end of the snout. And so that would tentatively place it in the genus Aldrovandia.
Is that a shrimp
It's
down
gone.
there? You gonna do a snap zoom?
Thank you. Good idea. There, Chris. Oh, there's a shrimp there. Yeah, that looks like a nematocarcinid shrimp. The common inhabitants of the deep sea.
Close.
This one, often you see them swimming. This one is gently climbing up slope, like we are.
Bouncing with my toe here.
It's really peaceful walk.
Such fragile legs.
He has really long antennae and long appendages that, you know, at this depth where you can't see much,
I'm pretty far away.
Can imagine how useful they are to sense your environment where you're walking blindly.
So based on that first move, I'm estimating...
This is safe to say that this is the first light that shrimp or that fish has ever seen.
About 17 minutes.
Really? Okay.
Maybe the fish, unless they migrate
so
vertically
if we did
near
one
the
every
surface.
17 minutes, Sirius would never get
If
built
they
up.
are constrained to deep sea, then that would be true.
And that's a 20-meter move?
Yeah.
Okay.
So it would be longer for longer moves, of course.
Yeah. Well, let's keep doing that, and that'll be a good cautious distance as we approach this steeper
But I mean,
section.
with the light, I
That's
meant
moving
more
about
like
50%
bright
of the
light.
time,
There
so
is light
averaging
also
0.1.
generated
We have to
by
average
many
0.06.
animals in the deep ocean. So
it should
there's
be
much more tenuous
good.
light. So it's called bioluminescence, and many
so I'll
different
call in the next move
kinds
then.
of animals have evolved the
let's
ability
see.
to
So we know from
produce
high
light
pack we want to be
either
going 135
through their own physiology or metabolic processes,
sonar's
or
kinda
in
vague right
associations
now.
with
Yeah, I don't know how much
bioluminescent bacteria.
I trust that right now.
So there's a fish on
So do
the
we wanna
top
keep
left,
going
looks like another
170?
holosaur.
Go ahead, watch lead.
Yeah, I'll do another that way.
Okay, sounds good.
Yeah.
Sure.
But it'd be great to take a look at the
so
fish
where are we at
first
in that
before he
last move? Is
swims
it pretty
away.
much finished up?
Yeah,
Fish
On top of the rock,
on
left.
top of the rock on the left.
All right, I will not call in a ship move.
Okay.
Sirius should be stopped.
Okay. Zoom out video. Are we full wide? You see a fish?
Yeah, I think it's on my way. It was,
I think I'd like to have you go ahead and stow the swing arms if I'm going to get down into that
Yeah, I think we lost it.
little hole there. Yeah, let's just do all of them for now, and then we'll stay in safe mode, and then we can deploy them later if we need to. Okay, sorry, I didn't see the fish you were.
Yeah, it was
pointing
just
out.
in the corner of the screen and it swam.
Okay, I can't hear you right now.
It was on the top left corner of the screen and it just swam quickly after that.
Okay, I think I could. I think you're muted right now, but I
something
heard.
white over on the very right.
I think you can hear me now.
Yeah,
Sorry.
there we go.
Pan right,
And this could
something
be
white over
something
there.
really different here. It's right on the edge of the screen there.
Oh yeah, is that a sponge?
Spongy.
Yeah, I actually thought it was a white translucent fish. I guess I'm in fish mode here.
And you're the sponge guy. Wow.
Looks like the pot belly sponge, but I don't see the similar attachment that the other ones we've seen.
Can you range down on the D2 sonar?
Yeah, that's a good idea.
Yeah, and boy, it kind of has a stalk and kind of doesn't. I guess my best guess would be Saccocalyx, but it's different than that other sponge that we imaged. But yeah, thank you very much. I guess we can call it a sponge fish then, huh?
And this tiny, small barnacle at the base of it, too.
There's a great rock right in the left-hand side of the screen.
Okay. All right, let's go ahead and zoom out.
Oh, no, you were there. Right above the lasers.
Oh, I'm going to move in closer. You're,
Roger.
in too far. And just help me watch the brow, co-pilot. That's pretty...
it's pretty close. You
pretty
want to
close there.
mini Zeus up to see the brow?
I'm not sure we'll be able to get back there, but we'll take a look. Go ahead.
So close, yet so far.
It's like driving a minivan into a...
Yeah.
Not sure. Oh, got some room actually.
A little bit. So you definitely want that particular rock because it was blocky and angular.
Yeah, I'm looking for things that don't look like balls of sediment and look like pillow fragments. And that one fit the bill, but
I don't know if I'm going to be able to...
I think you
that's
can
all right.
reach
We
it,
can just
isn't it?
keep looking
so?
around here.
I can try.
Yeah, your light bar is
We
safe.
could look at something else here as well. Might as well pan out. We'll leave that one be. This is going to be a bit tough, I think. Let's keep looking over to the right, if that's okay.
Oh yeah. You want to look to the right, watch lead?
Yeah, I mean this is a little precarious. I think. Leave that sponge fish alone.
That's fine with me.
Could have gotten a rock with a sponge on it.
Do any of these look like good candidates?
We zoom in a bit.
Go ahead and zoom in video.
Yeah, maybe to the left. There's a
Zoom out.
long block here. Right, the laser just a little bit to the left, tiny bit to the left. Right there. How's that look?
That longer one right there?
Yeah.
You want to zoom in? That
Does it wrap around or is it separate?
it's hard to say.
Looks.
I think it's separate. Huh?
I was gonna say it looks together.
The problem is that everything is ferromanganese encrusted, so it's hard to tell what we're looking at. There could be sediment underneath that ferromanganese cover or basaltic rock.
Do you want to go for it? Does that look like a good rock to you?
Maybe, how about the one, yeah, how about the one to the right there?
The one to the right. Zoom out a bit.
And just get the rock sampling over with.
Which one here? Go ahead and zoom in partial. Hold there.
Yeah, these look like nice specimens here.
Okay.
Poke around and see which ones are loose.
Okay, let's come out video. Yeah. Okay, let's zoom out.
There's a sponge on that one. Is that a sponge?
Looks
Santiago?
like it. Let's go full wide.
In the center?
Yeah, I'm sure Chris would object if we took that one.
Couldn't see.
Those are kind of tiny.
I don't hear any strong objections from Chris.
Shilling's getting kind of close. I think I'd be able to reach that. It's still pretty far.
I would say yes, but sometimes I've been tricked.
Yeah, you can certainly try it. It won't take long to go for it. I don't really want to get too much closer. You can see the arm. Yeah, let's do that.
So your brow is definitely clear now.
Yeah.
It's just the shilling.
Shilling, yeah.
I think it's close enough.
Okay, let's try it.
Just watch when you extend to keep an eye on it, keep it close so you don't just extend into the rock.
Right. Okay. Yes, I've got the pilot cam for the shilling.
Chris, if you're there, did you get an ID on that? Was it a sponge on the rock?
Yeah, you want to point the upper swing arms down.
I think they are down.
Are they down? They're
No.
up. Okay. All right, hydraulics are up. Everything looks enough down thrust. See what happens when the arm kicks. Make sure we don't move. Ready? Looks stable. Okay.
So these cobbles are clustering at the base of the overhang. As you move away from the overhang here, there are fewer cobbles. And that suggests to me that these cobbles came from the overhang, which increases the chances that they are basaltic in origin and not simply sediment.
That looks okay. Okay.
para mí también. Yeah, that's it's pretty small.
Yeah, do you want something bigger?
Yeah, maybe the ones over to the left there.
And which one here?
There's an elongate one.
The one we were looking at earlier?
Yeah, and then, yeah. Yeah, I don't know about this one. Maybe.
You want to zoom in?
Huh. Okay, yeah, if we see something towards...
Would you like this one? Let's go ahead and zoom out.
We'll just put this in the box and move on, ROV pilot.
So it's okay to put it in the starboard rock box with the others?
Sure thing.
Okay. Yeah, sure. Yeah, nice and slow. That's good. Sure. Yeah, that's good. I kind of bring it toward the back. There we go. It's tucked behind the other one. Sure.
So now that you're done, I've been noticing that that starboard side is always so dark when I try to put a bio or geo sample in there. So I've been having the co-pilot bring the uppers in like they are and then slowly moving them up and down until it lights up.
Okay. You sound
just
All right.
I don't know.
Thank you, RV pilots.
Is that better?
RV team.
Okay.
Anyway, if you found it dark, I always find it dark. And
So
I try
we're hoping
play
to
with it.
target 3 seamounts on the eastern side of the Simone EZ. This is one of
backing
them.
up.
And we're not going to get to the other 2 because severe weather chased us away. In fact, the storm had a name. It was Tropical Cyclone Bart, I believe.
So sorry, can you repeat what you were saying last?
Oh, I was just saying I find
we
that
are
starboard
currently
rock
in the
box
Samoan
and
region during
bio box to
hurricane
be
season.
dark a lot of the
so
time.
We'll be,
especially if the swing arms are out, the uppers. So if you bring it in and move it up and down until it lights,
dodging
until it just shines
storms
brightly in there.
Right inside. Yeah.
Same with the temp probe, but sometimes it's dark.
Yeah. That's a good idea. Yeah.
for much of the time during this expedition.
Okay. All right. Is that? Did you call? No, not yet. No move yet.
I can call it in now.
Let's go ahead and call it in.
Certainly, Cirrus has stopped.
Yep.
So we'll do. Yeah, I guess we'll do 175 again.
Okay.
Until the slope really solidifies.
I should correct myself. Hurricanes are called cyclones in this neck of the woods.
Santiago and colleagues, did you realize when you collected that rock how sticky that sediment is? how it's
Bridge nav.
not really coming off when turning and moving the rock. So I think in addition to the sediment, you are also
Could we get a
collecting a
ship
lot
move?
of bacteria
20 meters,
that form a
bearing
biofilm
175
and
degrees at
are holding
0.2
these
knots.
little sediment pieces together. Quite interesting, actually, to see that as well.
Yeah, unfortunately, Santiago
copper
just
bridge.
stepped out to grab a bite to eat. But yeah, thank you for your observation.
You can zoom in to lose the skids video if you want. There we go. Yeah, Carl, can you say a few words?
Okay, I dial in a little later, maybe
So
when
the
he
ship
is
move
back.
just got underway.
Okay, that's better. Yeah, there was some weird distortion
Thanks.
for a few seconds. Sounds fine now.
So I was beginning to think we wouldn't be able to dive on any of these 3 seamounts, and I started to refer to them as the Samoan Triangle, after its Bermudan counterpart.
Do you want to try switching the frequencies? I mean, I'd rather have a good, honestly, a good serious return than a D2.
Yeah, I'll play with it.
Oh, really? Oh, so this is better than what it... Okay. Okay.
This is a nice
go ahead and come
outcrop
down a
composed
bit.
of
I think we're pretty
nice
safe
pillow
here.
basalt structures that have been covered with ferromanganese. These ferromanganese deposits look very thick and developed.
Yeah, that seems to show less actually when I switch them.
Okay. I'll keep them the way they were then.
I think it's showing the terrain, and once we get steeper, it'll solidify.
It'll be harder returns.
Yeah, because I mean these are, you know, there's 2, 3, 4 clusters of rock up ahead,
right,
ranging. You know, this is the bottom.
Yeah,
a bunch of rocks.
So these ferromanganese deposits are very rich and economically important elements, including the rare earths and cobalt and so forth that are important in industry. The rare earths in particular are important in high-tech industry.
I'd ranged out to see.
There's been a lot of discussion about mining these ferromanganese deposits on the seafloor. Of course, major difficulty with that is that
so we expect
these
it to
deposits
start
are
solidifying about 30
very
degrees
deep, several
Celsius
kilometers
port,
deep. So right now we're
kind of looks like it is maybe.
we're nearly 3,000 meters depth, so 2 miles deep.
So we'll check it out
and
before the
there's
next move.
Significant expense associated with transporting anything from the deep ocean to the surface.
Well, in that case, let's zoom in on this creature on the rock. Go ahead and zoom in. Hold there. And zoom in tighter.
So this is a different kind of anemone.
Hold there.
Yeah, I don't think we've seen this one before. You can see it's different from some of the cup corals we observed a couple yesterday's dive, because these ones don't have a calcareous solid skeleton. They're not that kind of stony corals. These are anemones. They're all soft body.
Yeah, let's zoom out.
They don't have any hard structures in their bodies.
Thank you.
And continue moving along and climbing up along this steep wall.
So 30 degree support was according to HiPAC.
Yes. Western wall of the northwestern ridge on the slopes of Seamount D.
Starboard.
So just for your reference, this is the southernmost and easternmost dive that we will do in this expedition in American Samoa.
Copy that, Bridge.
We continue to see an
Co-pilot, do you see any movement yet?
Diversity of substrates here, so there are some large boulders that often have sessile animals, like there's a sponge coming into view on the left. And we could take a quick zoom, pilot.
Sure, go ahead and zoom in.
We've seen larger than these boulders roll down a hill. They have a lot of corals and sponges attached to them.
The ship started at 32, so about 5 or 6 minutes, yeah.
Okay, zoom in. Hold there.
This is another glass sponge, hexactinellate sponge. Has those fiber you can see actually inside the barrel of the sponge in the center. You can see the fibers, so those are silica fibers.
Santiago, I can hear you are back from your short break. I mentioned a little earlier that, I don't know if you saw when you collected the previous rock, that the sediment is really sticking also to the rocks. It comes off in like clouds, so it looks like you're also collecting quite some bacteria, biofilm that hold all these sediment pieces together.
Yeah, that's a good observation, Matthias. There's definitely, we know that there are bacterial communities living in the sediment, as well as small animals that we refer to as myofauna, very small animals, and also macrofauna. So there's a whole diversity, there's another ecosystem in those sediments. Right now we're seeing in the view a polychaete worm, the serpentopelagic. Normally seen attached to the seafloor, but they can also be counter swimming above the seafloor. But going back to your question, Matthias, or your comment, there is actually a large diversity of animals that live in the sediments, and this scientists and biologists that dedicate all their efforts in understanding the diversity and structure of the communities in those sediments. So there is both a microbiological and more animal component to basically the processing of the sediments. So some of those sediments have a load of organic matter that animals and bacteria take advantage of. So it's this particular organic matter that sinks from the surface and accumulates as sediments.
Got any more zoom?
And you're right that there can be some biological matrices that can hold together sediments and can make them sticky. This process is called bioturbation. So there's basically the mixing or disturbance of the sediments by animals as well that may lay their footprint on the sediments. So it's a combination of things, both microbiological and also animal activities that can modify the sediment.
Looks a little dark video.
So it's a mix of inorganic particles as well as organic materials that make up sediments. In this particular seamount, it's most likely that most of the sediments are coming from biological sources, since there are no continents or continental margins nearby. where there could be a lot of erosion that brings a lot of this sediment material.
Would just be cool to get a sample, you know.
Definitely. Fortunately, we don't have the right tools in this vehicle to get a sample. You need a sediment core, we call it, like a hollow tube that you can stick into the sediment and collect samples. But we don't have such tool on board.
Feature here.
Big rock.
So we're limited to collecting animals that we can, or specimens like rocks and animals that we can collect with the claws, or we also have a scoop sampler on
yeah,
board.
I was just noticing your sonar was kind of
But you have a shovel, or can you not?
like 20
I mean, I
degrees
saw
to port.
yesterday that you have a shovel to get like rocks. Can you not like scoop up a little?
That move? I mean,
surface of
so it should
the
be
sediment
stopping
and put it in one of the boxes?
very soon.
Just climb up and we'll take a look.
Yeah,
I stopped his camera still, and I've been
we
watching.
could do that. The problem is that there will be
coming back.
very disturbed. So one of the things you want to do when you collect sediments is to basically keep them.
That'd be great. I'm backing up.
The layers of the sediment, try to preserve those layers because there's a steep gradient of differentiation between what the surface of the sediments are and inside the sediments. So using a scoop is usually not a tool that we use to collect sediments because basically homogenizes the whole sample and it also mixes it with seawater that is above. So it makes it a little bit more difficult, or much more difficult, to make useful quantifications of the different parameters, biological and also chemical parameters of the sediments, and also the grain size, etc. So on top of this outcrop, we continue to see more sponges, as well as some large corals.
Okay, good. Thank you.
Yeah, thank you, Matthias, and those are good questions. Definitely things that we can consider for the future.
Both of them rotated up all the way now? Should be.
Several more of these small stock Demospongia, and this is Iridogorgia on the right, golden coral. Nice. And actually in the center, Pilot, we can want to take a look at that coral in the center.
We can do that.
We actually...
the move should be finishing up soon. We probably have some swing.
I think the swing almost stopped.
Oh, really? Okay. Good.
So actually, I've received a lot of feedback from folks on shore. That there's a couple of deep sea coral specialists, particularly an expert in octocorals, Phil Aldersale in Australia, and he has been sending me some messages indicating that this coral in the center, the one we be calling a Chrysogorgid,
go full wide video
in fact may be a completely different genus. Potentially be the genus Ricea, and that's actually in a completely different family, Eliselidae, and he says he's never seen one at these depths, or it's not something has ever seen. So there's been a request by him, as also other coral biologists on shore, including Les Wilding and Scott France, to support a possible collection of this specimen. So I wanted to also get your comments or input on this for maybe the people on the line, maybe in Hawaii and other places. I also support the idea that this might be something novel and actually a family of corals that we normally don't encounter in the deep sea, not that common in the deep sea, and definitely a novel observation for these depths.
Santiago, sorry, I stepped out to get some lunch since we're not allowed to eat in the ECC. And are you talking about the coral, since I just came back in, are you talking about the coral that's in view right now?
Correct.
Sure.
Yeah, although this particular one might be a different kind, actually. Just looking at the pictures, and the one we observed earlier had the branches coming out basically perpendicular, completely perpendicular to the axis, and this one looks more bushy, more like a Chrysogorgid to me. So I think this might be something different.
You want to zoom in? We'll give him a better view here.
From the one that
go ahead and hold there.
We have identified as potentially completely different family.
Give it more zoom if you got it.
Yeah, I'm not sure about this one. This might be a
Pilot,
Chrysogorgia.
arranged out your sonar.
I will look at it closer and looking at the branching patterns. It looks different. So maybe I will suggest we hold that collection for another specimen that maybe we'll be more sure that is the right thing, unless I get some rapid input from folks on shore. So I was mentioning that this is I just got this information from Phil Aldersdale, who's an expert core taxonomist in Australia, as well as from Les Watling in Hawaii and Scott France in the University of Louisiana.
So this is a no for a collection?
Yeah, I think we'll hold on this one. I'm not. This looks different to me, not like the coral that we are targeting.
Okay.
But it's a new observation nonetheless that is valuable. You see as a small mysis shrimp on that as an associate.
Did you want to look at the other coral below it?
That looked like a whip, correct? Oh, the Iridogorgia? I think we can. I mean, this is a really beautiful coral that I always like to see, but
get a ship move called in.
Yeah, if you start centering up, I'll start to call it in. So
if we have some time to spare here, we can take a closer look.
I think Sirius sonar looks pretty good.
Yeah.
There's kind of stuff off to port and starboard. So
go ahead and zoom in.
Yeah, there's just a collection of boulders all ahead. I don't know. I could do anywhere between 150 and 170.
I'm going to do 160. Split the difference.
It's good math. You like that, co-pilot?
It's okay. I'll come up up and over this thing. That should work.
Yeah, I don't see any associates on this Iridogorgia. Either we didn't observe any associates on the previous one.
Okay, sounds good.
From the shallower dives, when we encounter this
I'll call it in once you jump up and
kind
over.
of coral, we were observing squat lobsters associated with it here in Samoa, but the last couple
about
colonies we observed
when
here,
the ship starts to Sirius starts, and then after
2,958 meters and deeper don't have any evident associates.
Bridge nav. Could I get a ship move 20 meters bearing 160 degrees at 0.2 knots? Good copy, Bridge. Co-pilot, you mind if I put your sonar back to 50?
I don't suppose he could. Well, I saw an unbranched octocoral again. I guess if we see one that's within range as we go up slope, I think a bunch of people would like to get another look once again at that unbranched bamboo or chrysogorgid or whatever it is.
Oh, the whip one? The whip bamboo?
Yeah, there's some discussion. Some people thought, as I did when I first saw it, that it could be Radicipes, which is a genus of Chrysogorgia.
Yeah, no.
We just, now that some other people are now on the event log and watching, it'd be good if some other, bunch of people weighed in on it.
Sure. All the whips that we have imaged today so far have been bamboos.
Descend down to the bottom.
I've seen the internodes, or the nodes rather. But definitely we can take another look whenever we see another one.
Well, even that, it was a really interesting bamboo, and probably Scott and Les, I don't think they saw first one. I think both of them might be really interested to see the close-up now that they're on.
Sure.
Is that something that's
Keep an
in
eye
view
out.
now, or something we passed?
No, something we passed by several meters.
Okay.
So we probably see it again as we continue moving upslope.
Sure. Yeah, go for it. Tether's good. Sure. Another Holothurian. Go ahead and zoom in. Hold there.
So this is a different kind of Holothurian, and also with a sail. This is actually, I think, the same kind we observed a couple of days ago. You see the sail has, I don't want to call it branching, but it has two lobes, so the two pointy tips at the end. The one we imaged earlier was different coloration, was more translucent, and it had 4 of those tips on the sail, so that part of the body that just projects out of the main.
Okay, let's zoom out. Stirred up some sediment there.
It's really amazing animals. Really strange.
So you called that last move in at, what, 49?
The ship started at 50:30, so it should be moving 1 or 2 minutes.
Okay.
It'll take about 5 minutes to move.
So Steve Auschwitz and I both thought that could be something called Cycropodes in the family Cycropodidae, and it's also in the Okeanos animal guide.
Coming up.
If it looked like to both of us, just to give a name to it.
Okay.
So I like these short moves with you way out in front.
Yeah, I agree.
It is kind of solidifying and serious. You can see,
yeah,
like 30 meters out, there's a big boulder with a shadow.
Yeah, it was
actually just came in real clear there. Yeah.
You can see it's a little more rocky above.
Maybe.
Looks like you're moving, huh? See that boulder below us moving down off the bottom of the screen?
Yeah, it looks like it. Copy that, bridge.
Another Holothurian over there, I think. Or is that a rock with something on top of it?
I'm not sure. It's different.
Do a partial zoom. Oh, it looks like maybe a coral, actually. Huh? Yeah.
Oh yeah, that was the shadow.
Yeah, Pilot, this is the one people on
Okay.
shore are interested in re-imaging.
Okay. Well, we got plenty of time for that. So I'm going to push out
Excellent.
Andy and sit down. Just let me know when start getting over the top of
Yeah,
it.
so here is Chris, again the whip that we observed this dive that you mentioned. The folks on shore wanted to take another look at.
Standby video.
Yeah,
Come in
that's
closer.
the one I think.
Yeah, it's the only whip we've seen so far in this dive, or the kind of whip.
Okay, go ahead and come in.
And we can pan down. Oh, let's see a mysis shrimp right there. Take a closer look towards the base.
We get a rewind to look at that.
So this one
Go ahead and zoom in.
doesn't actually have the nodes that I saw in the earlier
Is that
ones.
full zoom? Oh, okay.
I think there's a node right. Pilot, can you stop the camera? Or tilt a little bit down. Right at the bottom of the screen. Right there. I think I see a node right there.
Up in the upper left too.
Yeah.
I see what you're talking about, Santiago.
Yeah. Yeah. So they're
Just
not
kind of
as close together.
More rocky rubble field up ahead.
And this might be. I think this is on a loose rock. I don't know if you pilots can zoom out.
You can zoom out.
Wonder if where it's attached is a loose rock or not.
Yeah, that'd be. You can count this as a. Maybe count this as a fourth geo sample.
So that.
Oh.
So what if there's interest from shore? Maybe we can attempt to collect this.
It's a fourth geo sample.
How'd you get it?
I could turn it upside down
and let it crush itself.
Yeah, this is we can try to attempt to collect this bamboo with this interest.
It would definitely crush it, but we could.
Well, it's either that or you set it up right and then try
Box
to
it.
stuff it, which would be, I think, upside down might work.
Well, I would
it
support collecting it.
Swing into the corner.
I don't know.
of the
I'm
rock
pretty
would
confused by it.
might
Both
work.
Scott and Les thought it could be the chrysogorgid, the Raticides. But I saw what you were looking at a little bit better, even further up on the stem.
The tip would
did
poke
look
into
like
a
a
corner
node
and
to
it might
me.
just curl itself up.
Yeah, and we've observed
think it
before.
definitely
We
goes
got
upside
some
down or
other
sideways
images
or
with the notes
something.
as well
So I think
earlier.
Just putting it in, it's going to be real.
But we
springy
can, if there's interest and you think there's thoughts
one
of
of the port
folks
boxes is
on
open,
shore
right?
that it might be something
Port outer,
unusual
which
or
is
new, we
larger,
can
I would do port
attempt the
outer.
collection here and maybe try to get the rock that is attached to it done.
All right, what's the call watch lead?
Looks like you're getting support for a collection.
Seems like we want to go ahead and
with or without the rock.
How you
I
doing,
think
Andy?
it's probably a good
Happy?
specimen, no matter what.
Roger, so we'll attempt the collection of this rock coral.
There's a coral with a
I think you
rockfish
could leave it
associated.
there. That's fine. Yeah, that's fine, and I'll tilt down a bit.
And I guess if the rock's stuck, it's an easy
Maybe a
clip anyway.
shadow.
Yeah, exactly.
Work out.
A shadow rock.
Okay, hydraulics are up. Okay. Everything looks stable. Comfortable. Yes, please. That'd be great.
It is out there, but I think you can reach it.
Yeah, I think I can. All right.
Oh yeah, that's plenty close.
Ooh. That's a much bigger rock. Oh, no, maybe not.
Just a lot of mud.
Oh, wow. That is a beautiful pillow fragment attached to the coral.
That's pretty big, huh? Carl, what do you think?
I think it'll fit. The trick will be getting the
Would that
coral
even
to
fit in the
fit.
bio box, or would it be better to snip off the coral and put that in a bio box and the rock in a rock box or something?
Yeah, I was just asking that to the pilots, and they think it'll fit in the bio box.
Squish it a little bit.
That sediment is coming off. It's amazing. This sediment has never been disturbed before, so you're looking at the accumulation of.
They want the sediment, actually.
Yeah. Oh, okay.
Hundreds of thousands of years of sediment, or
guy
longer,
talking about
likely
it earlier.
millions of years of sediment. Just
I'd really like some sediment.
see them trying
to wash it a little bit.
Shake off that sediment. That's a beautiful pillow fragment. Grab a little rock, and there's a lot more of it buried under the mud.
I'm thinking maybe I should re-grab it. What do you think? Or actually, well,
I think
you
could go past it, put the whip in, and then bring it closer to you.
You grab the rock and roll, and then we had some rock and roll.
And then over to the left a bit. And then that's good.
Without.
And then might have to come up for the drawer, huh? Unless maybe if I wing in.
Yeah.
Yeah. You want to? Yeah. Let's try it. What's in the port box? Nothing that'll swim away, right?
There's something important there. I don't know what it is.
It's a piece of coral, pilot.
Okay.
So.
Yeah. Okay. We can just go and
It should
open
be.
it.
It's like a full coral colony actually, but it's not that big. So it'll be fine. And it's not gonna swim away. That's what I'm saying.
Well, that fish rock might swim away.
Yeah, I'm getting pushed a bit. You're fine. I'll just come up a bit. Let's see. Let's do
Yes, I think it's the port outward is the one that is empty.
Just keep an eye on, actually if you put Sirius on monitor 12, so we don't have the
I see it over there.
Okay.
Yeah.
All right.
I think I'm not keeping an eye on Sirius.
Oh, not. Just be. I could just look over too, but I trust. Okay. Up a little more. Let's do that. There we go. Okay. Let's go ahead and open the drawer.
Which box is this, Santiago?
I think it probably will go on the port outboard. Most likely.
Okay. Ooh, that's pretty big.
A little closer. It's going to be hard to get the tip of the coral in there.
Yeah. I could drop it in and snip any part of the coral that's sticking out potentially.
Is there a way to get the tip in there? No, it's kind of far away. Huh?
Yeah, well, maybe. So keep an eye on the jaws to the shilling there, co-pilot.
So Santiago, you have 4 bio boxes, correct?
Correct. There's really 2 bio boxes separated by each one separated with
That's nice.
So for the potential
plastic
of 4 distinct bio samples.
Correct.
Including a fish rock.
fish rock.
Not working?
Nope.
Yeah, maybe I'll have to set it down and snip it and do 2 separate.
Yeah.
Or I meant set it on the ground, but
Oh, that looks like it should be a perfect
whatever.
fit.
Oh, you have it.
Yep.
There you go.
Wow. Nice catch.
Oh, I just nicked the shilling. Sorry, Roland.
Look at that. These guys are really skilled. This RV team. They can use this claw to do really fine tasks. And I understand it's much, much more difficult than they make it look.
It's still pretty long, huh?
I think that'll do it.
Can you stuff it down a little?
How does that look, Santiago?
Actually, you should step back a little.
Looks good. Most of it is in the bio box.
Do you want me to do it? Or?
Get it.
It's just a little
careful, the joystick isn't really
tip
mapped
just
to
sticking out.
Right. Just go back a bit. Just come up and out. Let's go ahead and just close it up. I mean, I think it's mostly in there.
So, pilot, I just warn you shouldn't move too much. The coral on the right.
Yeah.
Do you see it is sticking out?
Oh yeah. It's in the box. Okay, let's close it up.
Yeah, they're both in the bio box. All right, great job, guys. That was the bamboo whip went into the port outward bio box and was collected at that 2,952 meters.
Well, actually, are you marking that as geo or bio?
We marked it geo.
Thanks.
Okay. That was great. Now we can continue our track.
All right.
So we're at that 2,952 meters, ascending on the western face of a ridge on the northwestern side of Seamount D. So this is a seamount that we just mapped in this expedition. It doesn't have a name, so we just call it Seamount D for this dive, and we refer to it as Seamount D. So we continue to see this diversity in morphology of the seafloor.
We'll
Come across some really large
steeper, we'll put 'em in again.
boulders, look like the attached boulder that roll down the hill, as well as these patch areas of sediment cover, as well as some cobbles and smaller pebbles. None of these look as rounded like the ones we've seen in other deep seamounts that seem to be much more encrusted with iron manganese. Don't you think, Matt?
Yeah, these you see a lot more obvious billow fragments here.
Yeah. More angular.
Okay, so
rocks
train should be steepening up here.
Thanks, Andy.
Also seen a diversity of corals.
For you guys to get comfortably lined up and pick
associated
a
corals and sponges attached to the rocks. Species of holosaur fish, and a couple different kinds of sea cucumbers.
Go ahead, Bridge.
Tilt up a little bit.
We might suggest
push ahead.
A Samoan word beginning with the letter D for this seamount.
That sounds
Instead
good,
of calling
Bridge.
it Seamount D.
That's a good idea.
And that was 160 again?
Maybe we can get some suggestions during this dive.
Oh, I haven't called in yet. They're doing a heading change.
Okay, gotcha. So we got some time.
Yeah. Yeah, his
There's
camera's
a Samoan
66, so you can move out ahead.
Fellow on tow who
Call
I
him a move in a
became
minute.
friends with during many visits to the island.
But
a fellow by the name of Dino.
It looks
Samoan
more
name,
like it's tending to
D-I-N-O.
starboard on Sirius because the port side of the sonar is kind of empty.
Yeah.
Actually, take that back.
Right.
Yeah, 150 seems pretty good. I think eventually it'll tend that way, yeah.
Maybe another overhang here. Can't quite tell. I'm gonna come up. Yeah.
It's interesting. Yeah, so that's the stuff to the starboard of the Sirius
So
sonar.
here.
Abundance of
Come up
pillow
over that,
fragments
and then
below
there's
this
a big one
overhang.
ahead in the Sirius sonar.
Yeah, it's a really another of those really large boulders that likely round roll downhill. There's a stock sponge of the seem to be a Calophacus. You could get a snap zoom on that sponge,
Sure.
pilot.
Go ahead and zoom in. Hold there.
It does look to me like a Calophacus.
Okay, and zoom out.
Yeah, thank you, pilot. So there wasn't that much attached to that rock there. Just wondering maybe that was some more recent roll down the hill for that boulder, but it's hard to.
It's hard to tell. Yeah, but we have observed some really large blocks today. They look like several meters in diameter at least that aren't attached to the substrate that appeared to have rolled.
Yeah, and most of them had
down
a
the hill.
high abundance of corals and sponges attached to them.
Yeah.
Except for this one, I only saw one sponge on it.
Thanks, Bridge. Okay. So the heading change is complete. So this was the move that you prepped, 20 meters, 150?
Yeah, I was thinking, since I am already pretty far out, maybe we should do a 30-meter move.
Okay.
Looks pretty flat here.
Sure.
There's something in Sirius sonar about 40 meters away, you can see.
Yeah.
So maybe we'll get us me to there, and that'll be something interesting.
Okay. So toward 150.
So we're seeing here
we'll call it
top of these outcrops.
Bridge, this is nav.
An larger area of sediment cover.
We'd like to request a move
with ripples
of 30 meters. That's 30 meters, bearing
strong
150 at 0.2 knots.
Or strong enough to make ripples in the sediment.
That's correct.
Okay, proceeding ahead.
Thanks, Bridge.
The sediment cover continues to be extensive on this slope. I'm guessing this slope is going to be around 30 degrees, Santiago?
Yeah, it could be here locally, but I think we're in the general average slope of this ridge side that we identified with the multibeam bathymetry. I think it was a little bit between 30 and 40, if I remember correctly. So it's natural to see some variability at the smaller scale. Oh, Pilot, I think that coral in the center is the one we were targeting for collection earlier.
Okay. Co-pilot, will you come down a few meters? Give me a little more tether.
We can take a closer look.
This is the one we just collected also, or a different one?
No, this is a different one.
Okay.
We were looking for this one, and we just happened to encounter the other one that also was of interest, and it happened to be nicely attached on a loose rock, which is rare to observe here.
I suppose Matt wants the rock it's on too.
If possible, yeah. If you could drop 1,000 pounds of ballast, you just might be able to bring it up.
Just waiting here a moment. Sirius is underway, and a couple minutes I'll have enough tether to get over there.
Roger.
In the meantime, video, will you zoom center screen?
Oh, that's a shrimp, yeah.
So what is this we're looking at, Santiago? There's a coral above this shrimp.
Over there.
Yeah, this is an hematochrystinid shrimp. You can see we saw one earlier that was walking on the sediment.
Yeah.
This one is being just static on this rock.
Yeah, the really long legs, like the daddy long legs I'd see as a kid in Montana. I'm going to call this the daddy long legs shrimp.
Yeah, they certainly have long legs. Pilot, if you want to tilt the camera, I see this little starfish or Opheroid on the left. So
can you go back in video?
To the left of, no, not in this rock, and this part of the rock, but in the left one. I'll say right below the left bit laser.
Okay.
Right there.
Video. I see it now.
Yeah.
Oh yeah.
There.
Yeah, it's an Opheroid, brittle star.
I think you saw an Opheroid earlier when I'd stepped out. Opheroids you've seen thus far have
don't
been
recall.
associated with the corals.
Oh, the clear translucent?
Go a little
Yeah,
tighter
yeah.
video.
It's a different kind. This is trying to remember the name, but this kind is more usually associated with rocks rather than the corals.
Guys, could we take a look at that leggy coral after this brittle star that's on the top of this rock? Because there's some of us are wondering whether it could be the one that Scott and Les and others were interested in. It sort of looks like it, but can't really tell.
Yeah, Chris, I think it is. That's one I was indicating the pilots we wanted to likely collect. Just waiting for Sirius to get a little bit closer. Right now, we're the stretch with the tether, so we're just heading there, zooming in other animals as we reach to it. So this is the coral with sparse branching that we had been earlier identifying as chrysogorgid octocoral. However, received some feedback from coral expert and taxonomist on shore from Australia.
There's a feature probably 10 meters.
That is suggests that this is a completely different kind of coral. It's
D2.
Copy that.
That was a 30
likely in
meter
the genus Ricea.
Thanks, Bridge.
Okay, video can go in.
So Sirius is probably starting to move about now.
So
yeah, Copilot, you can bring your delta back up now that I've tethered.
this is considered.
Hopefully that.
If it is, this is the case. That is in the genus Ricea. This is a different family of octocorals. So it is elicellid, and it's a rare observation since we know that these corals mostly mostly from shallow water. So there's a great interest in collecting these corals since it's so unusual. So this is the one I was indicating earlier that we wanted to collect.
Yeah, I think we are. Okay, we go wide video. You guys feeling comfortable?
Yeah,
if
it's
you were to back the ship up 10 meters.
Yeah, I might do that. Yeah, I'm gonna go ahead and do that. Oh, if you do that too much, it'll tug on his tail as he's trying to sample. Oh, really?
Okay, I think D2 is pretty stable there. We cannot put the box out.
So we did observe this same morphology, or the same morphospecies at this depth range on Utu Seamount further north in American Samoa in dive 6 that we didn't collect it then. So this is a great opportunity to really take a closer look at a specimen and identify what it is.
Yes.
At this point, we are debating whether even what family it belongs to.
Yeah, can we please have another
information
ship move of
that
10
we
meters? That's range
have is correct.
meters.
We might be seeing a
bearing
really unique example of a member of this family, Helicellidae, living in deep waters.
I apologize. 330 at 0.2 knots.
About current that is 2,042 meters.
That's correct. Thank you.
Okay, we rotate the mini Zeus to be kind of square.
Thanks, Bridge.
Video, will you go in on Mini Zeus and try to bring this coral out? I don't really see it. I see it on share. Yeah. There it is. Thank you.
So we'll see. We'll send
Blending in
some
with the
images.
there, I didn't even see it.
Close-up images of the collection
Video,
to.
you can actually go back to where you were.
Our friend Phil Aldersdale in Australia, just an expert taxonomist in corals, particularly more soft-bodied corals, and maybe he can give us his opinion, as well as hopefully getting a chance to look at the specimen that will be deposited at the Smithsonian Institution, the National Museum of Natural History. That's the ultimate repository for the biological samples that we collect in this expedition, to make them.
No.
Oh, it's brittle.
This was brittle. That was unexpected.
Wow. Yeah, I wasn't expecting it to be brittle either.
Well, I have a few inches in my jaw. I should definitely put that in the box.
Yeah, if you have a piece in your jaw, maybe just put it in the bio box for now, before we
actually
try to get a bigger piece.
Actually, yeah, I don't have any.
Yeah, I don't see any. Do you?
There must be a little bit inside the Tygon, but
that's interesting. I was not expecting it to be brittle at all.
no. Okay, I'm going to tilt down on main HD.
So.
No, that's okay.
So, Chrysogorgids, that's what we've been calling it before, belonging to that family, usually have a protonation skeleton that usually very flexible.
Yeah, so watch it drifted down here.
Thanks, Bridge.
Yeah, I see it. Is that still reachable with the arm?
I'll give it a try, just given how it kind of disintegrated there when I touched it. I'm pretty sure if I touch it again, it will.
Yeah. So usually we see these more brittle characteristics in things like bamboo corals. They have a harder skeleton.
Yeah, will you tilt Mini Zeus down, co-pilot? Are you guys happy with Sirius?
Yeah, the ship moved 10 meters backward, just completed. Okay. So I think we'll be comfortable. There is a pretty large feature up ahead, but I think we'll be fine for now.
That's probably okay, video, will you go in on Mini Zeus?
Well, this is really interesting. None of us were expecting this.
We're actually going to go for the, I think.
So we so brittle.
Well, actually, yeah, go in on Mini Zeus, see if there's enough polyps on the upper part, sorry, by the jaw.
On these relatively deep dives, Santiago, we never know if we're going to see much at all. And it's been one surprise after another for the biology. I landed in the real Garden of Eden.
Watch lead pilot.
Go ahead, pilot.
Yeah, there are, looks like maybe 8 polyps left on the stem here.
Roger.
I could do a second snip on this.
You think that's easier than grabbing the piece that already fell off?
Well, so you actually want the branching one that fell off, is that?
That'll be the most desirable, yeah.
Okay. All right, let's bring the Mini Zeus down to that.
Santiago, did you see how this thing was, disintegrating? You mentioned that was unexpected, but I think it also shows that down there is really no current. So, this is amazing. So there's complete calmness in the water. Otherwise, the thing would never grow to that size.
Yeah, that's definitely, we don't seem to have much of a current right now. However, we do have evidence by looking at the sediments that there are currents that go through this area, and that's because we see ripples in the currents. Those are characteristic of evidence of
do you
significant
think, Dan? What do you
currents passing through this area. So
I'd
that
probably
just try to
there can be variability over time of speed of currents, and given the morphology of this place where we are, it's more like a valley. It's likely that there are actual currents being or increased circulation velocity of water being canalized in this area.
Really close here.
But yeah, it's interesting observation you make, Matthias, that right now we don't seem to have any current at all here.
I'm going to try to...
Yeah, it's very interesting. I mean, it's amazing. So, I mean, on Earth you have these winds and whatever, and the trees are all kind of like adapted to that, but that coral apparently is not at all. So, I mean, it's just breaking apart if there would be any current. Anyway, I also sent an email to all guys in our department, so I hope some people will tune in. And I heard from some back that they are also following online. So, well, amazing opportunity. Again, thanks so much.
Yeah, thanks to you. It's great to have you in the line. Oh, this is a very delicate job right now. Wow.
Oh, very nice.
Okay. Wide on both cameras.
Picking daisies with an 800-pound force claw. This is a lot of skill involved here.
Okay, I think to open the boxes I'm going to have to take off. So you ready to open the starboard box? Stand by, I'll just get into autos and then you can open it. Should hold. Okay, you can open it up.
Hello.
Hello. Who's joining us?
Hi, Santiago. Is it Santiago?
Yeah.
Nice.
Hey, this is Joe Bragg from Lehigh.
Oh, hey, how are you?
Dan,
good. How are you?
Keep an eye on my,
great. Thanks for joining us.
Look fine for now.
Oh yeah, I was just watching your live stream there on the ocean floor. It's pretty cool stuff, man.
Yeah, it's definitely, we've seen really amazing observations. Right now we're collecting a coral that has us puzzling here and interacting with, several of the experts in these groups, and none of us can agree on what this coral may be, not even to the family level.
Huh.
So it would be a really great sample to characterize what this may be. It's very likely this is going to be a new species.
It's down
I think.
in the bottom there. You can close it up.
It's definitely a unique observation and collection that we're doing right now.
So how do you name a new species of coral, Santiago?
Well, first, more importantly, you need a sample like the one we collected, and first you need to compare its morphology to what's known. It's important also to note where it was collected, what depth, and the geographic region. We usually look at the skeletons, the large skeletons, as well as the microscopic skeletons that are found inside the polyps and in the soft tissues. And one of the things that's been more important in recent years is definitely do a phylogenetic analysis. So looking at a few genes, or ideally large parts of the genome, that give us a much better, accurate identification of the relationships of this coral to other species, and from there we can build on and characterize the depth range and
the only thing that was tricky was
try to address more interesting questions like how it's adapted to live at these depths and
close them again. So
what are
hitting the inside there.
characteristics that determine its morphology and the features that it has. So why is it so brittle, for example? Matthias was pointing out, there's no currents right now, but we were seeing the sediments as ripples here, so they do see some currents. So it must be flexible enough not to break with the currents. But
just push out,
as we saw, it was very brittle. So it's interesting just learning how these morphologies and the structures are being generated in these animals.
I'm going to find something up here.
Yeah.
So that was the sample specimen 6. So this is a biosample that Pilot, can you remind me if that was in the inner or outer starboard?
That was inner starboard.
Inner starboard biobox. And that was collected at depth to 2,930 meters.
So just come up and make sure there's no overhangs here.
So you've had the opportunity to describe new species and name them, Santiago.
Actually, no.
That's correct.
I
Yeah.
was just going to do the minis yourself.
And that was the process that I just described. Yeah. So
yeah,
actually,
it's pretty slow.
So how did you pick the name eventually?
Well, there's different ways you can name things, different species.
Yeah, that looks good.
You can. There's a few rules. One of the rules is you can't name it after yourself.
That makes sense.
That you can name it after other people.
Can you name it after the pilots who collect it?
We can, yeah.
Oh, nice.
Yeah, so the ROV pilots have a question for us. I don't know if this is being broadcast to everybody on shore. They'd like to know if we could name the species after them.
It's possible, but it will be...
ROV as Pilotus? ROV Pilotus Corrales?
It will be up to the person that describes the species. What name it gives them. Definitely, that's a possibility. I'm not an expert in the particular group. My focus has been mostly on the bubblegum corals. We have only seen one of those here, but still would like to take a look at this, definitely when it comes back.
So Carl McCletchy is our current ROV pilot, and I was thinking like a McCletchicus gigantus corellis.
Sounds perfect. All right, I think I'm pretty stretched out here. Should we do, I think 20 meters?
That was 3 words that you said, right? The name?
Yeah, I know that. This
right toward 145.
That might not be preferred.
Yeah, so we use a binomial system on nomenclature.
What about Homo sapiens sapiens?
Well, that's our subspecies, but
video quick zoom on this.
As far as we know, we don't have
I'm going to call it in.
octocorals to the level of subspecies. There might be some, but I'm not aware of it.
Ship move, please. 20 meters, bearing 145 at 0.2 knots.
So what we're seeing for the geologists out there, we're looking at ferromanganese incrustation here, and we have
that's correct.
A typical bubbly surface appearance, like lizard skin. This covering, this is an extremely common
one
morphology.
of those Polyopogon sponges, I think.
Netcams have the best lighting and view and color.
Yeah.
Can you repeat that genus name,
Yeah,
Chris?
they're nice little cameras.
Yeah, I think it's another one of the sponges in the genus Polyopogon that we've seen before. This is just a smaller one.
Thank you. There's some pilot. You see something clinging on the rock on the left.
Yeah.
Some.
There's a fish here. I'll just try to.
Oh yeah, let's get the fish
it's not in
first.
spot, but okay, video. If you start going in on the fish.
Is it a fish?
Or a worm? I'm not sure.
Or.
Yeah, it's just something.
Or. It didn't look like a fish from the distance, and
no,
now.
It's a worm, I think.
Is it a worm? Oh.
I'm not positive of what this is.
What's on the lower right? Some tentacles, or can we get a tight zoom on the lower right portion of this?
It's neutrally buoyant. You see, it's not.
Yeah.
Those tentacles, Santiago?
It looks like an arm of an Opheroid or something like that, but it has some. Yeah, you're right. It has some projections at the end. It might be a worm, but I've never seen something like that.
It's going away.
Anybody in the line?
What?
Have any idea what that could be?
Back to this coral.
Somebody threw out, Kevin threw out a possible ID in the event log. If you take a look, Santiago.
Thanks. Yeah, that's not familiar to me at all. There's something else also in center in front of us.
Good idea. Yeah, I'll stay here. So yeah, I see this bushy coral right ahead, and then the one of interest is just above.
Roger. Yeah, you can take a look at both of those.
This looks like it has the appearance of a dike almost. See this? This cross-cutting.
On the left?
Yeah, it's sloping from the lower left to the upper right. So elongate magmatic intrusion.
This looks like a dead sponge skeleton that is being colonized, perhaps, or likely by some hydrates.
Go bridge.
Yeah, I think that's what it is.
Yeah, I'd agree. It was some type of a base sponge, either a Walturia or Rhea or something like that, possibly in the Euplectellides.
Yeah, that's fine.
When it was alive. Now there's not a whole lot left of it.
And then you're going to bring your head to starboard.
Oh,
10
this
degrees.
is maybe one of those plurogorges that we were seeing, although it looks a little different to me. We'll find out in a second when we get a closer look.
So it looks like Sirius saw this giant pile of boulders and rubble, and now there's nothing. So
yeah.
Good. Yeah, I think so.
So that 20-meter move should be.
Yeah, the geology down here is spectacular. You can see lots of exposed pillow fragments. And some of the seamounts, these older seamounts we've observed in the last week, the ferromanganese incrustations are so extensive you can't even tell what's being covered up. You can't tell if you're looking at sediment or carbonate or basalt, but here you can see the pillow fragments scattered around very clearly. This is a veritable paradise for someone like me interested in looking at and collecting basaltic samples for age dating and geochemical analysis.
Also for corals.
Of course, for corals. Corals love these hard substrates.
Yeah, so this is another octocoral that we've been calling a plurogorgia within the family Chrysogorgiidae, and this is the kind that has dark skeleton. This is a nice view of the polyps, and you see some of the polyps have these extended tentacles, the ones in the center, and then to the left, there's some polyps that have the tentacles retracted. And again, an excellent example of the characteristics of an octocoral with 8 tentacles and the lateral projections from each tentacle, the pinnals that are characteristic of this group. Yeah, this is a very nice image here. And thank you, pilots. I think we can continue.
Okay, Vidya, if you just frame the specimen, we'll try to get the lasers on it. They are. I'll just tilt up.
Yeah, you should start to feel that 20-meter move now.
Okay, that's good. You can go wide whenever you have your shot video.
ROV nav.
Go ahead, watch lead.
How much time do we have remaining on bottom?
Sean had made notes. I think it was at 2 p.m. We had to leave.
2 p.m.
What's
Or
this on the left here,
2
Santiago?
UTC.
Coming into view.
Looks like a sea
15
cucumber.
minutes left.
Oh, pardon, could you repeat that? I
1
apologize.
hour and 15 minutes left.
1:15.
Roger.
It makes sense. An hour and a
So
half.
we got some more corals here, and a field of view. There is
Can go in on the cucumber.
Holothurian sea cucumber. So
Okay, hold there.
different species, a different kind from the ones we've seen with the sails, with the long sails.
Very different.
Mm-hmm.
There's numerous
You can go in tighter.
protrusions all over the body of
Thanks,
this
Rich.
organism. Is it difficult to sample these, Santiago?
They can swim,
Okay.
and they also have the body is it's a soft body animal. It's not impossible, but those are considerations.
Again, you can see the sediment that it's been consuming.
Yeah, with translucent.
Yeah.
Translucent body. The only thing that is definable structure is the digestive system, and it's filled with sediments. As you can see, these animals are deposit feeders, so they feed on the sediments that accumulate on top of the rocks on the seamount. They're common to see in sedimentary areas, although we've observed several species that are like to be on the rocks too, eating the small deposits of sediment that form in the cracks of the rocks and the depressions.
Done by now.
Okay, should be stable there. But yeah,
These animals can move over the seafloor slowly, but they can also swim. They usually you see them dropping the ballast of the gut contents before they take off. So they are obtaining energy through digesting organic materials or organic particles that are in the sediments or forming the sediments, as well as microorganisms. And possibly myofauna, small, very tiny animals that live in the sediment.
The local is not quite that.
So this is.
I think.
Can we take?
We can follow the local.
I think we want to image these two. There's a small coral here that looks like a small Iridogorgia.
Yeah, I see it.
The spiral coral. We've seen much larger individuals. And there's also stock sponge. This one looks different to me. It's very long, slender stalk and a small head.
You're happy
coal
where
head is
you're
not
at,
a real
88,
head.
160. Looks good to you? Okay.
I'll zoom on these. And then,
So you have just over an hour left of this dive.
Sounds good.
Santiago, and I don't know if there's interest. You can still get a couple more biological samples.
We'll see what people on short think. This.
It's not very good contrast. Let's see what you can do, video.
There's a tiny sponge for such a long stalk.
Right off the center.
Yeah, it is, and I thought it was. I think we observed some of these early in the previous dive. I was calling them Calophacus, but if I remember correctly, Chris Kelly corrected that this was indeed something else. So this does look like a juvenile Iridogorgia golden coral, like basically the same kind of those long, really long and tall spirals that are so beautiful. We observed one a few dives ago that it was more than 2 meters tall, a really large coral. In fact, Les Watling, a biologist in University of Hawaii and collaborators, published a paper suggesting that these corals may be the tallest octocorals in the deep sea.
Want to go in on the top then. Co-pilot, we tilt my camera.
Well, I still think it's a Calophacus of some kind. It just has got a really small head, as you noticed.
Yeah.
Unusually small head. But I still think it's at least in that genus.
Okay, thanks, Chris. Yeah, it is unusual to see such a long stalk and a small head. Usually see the proportions being not as biased toward the stalk.
Watchley, do you want a better view of these? I wasn't very well set up there.
I think, I don't know, Chris, are you happy with the imaging of the sponge, or you want to take another closer look?
No, that's okay. I think we can move on. No problem.
Okay, yeah, we can move on. I see another Pleurogorgia just in the top right part of the screen, and I see a large, likely possibly an Iridogorgia that is sticking up in the background. It's gonna come into view in a few seconds.
Oh, look at all these pillow basalts outcropping.
Oh no, it's not an Iridogorgia. It's the same kind that we just collected. That the octocoral, that is the mystery octocoral.
Huh.
Snap zoom here video.
That could be either Chrysogorgid or a debating also being Eliselidae or even a bamboo coral. At this point, we don't know.
What?
Maybe I can give you all an update once we get the sample on board and send you some pictures. That'll be more detail under the microscope.
Definitely a pretty significant feature we're climbing here. Looks like maybe it'll be more of a 45-degree rock instead of a series of cliffs.
So
it's about
20 meter rise over 30 meter run.
So this terrain looks different.
Yeah, this is starting to look more like a volcanic breccia or volcanic hyaloclastite or something. I don't see the clear pillow basalt structures here anymore. The sediment cover is greatly reduced compared to before.
Okay, I'm comfortable with another move.
There's another coral on the left.
I think 20 meters toward 160.
I think it's likely to be another one of those Pleurogorgia like corals.
Sure, yeah. Ooh, what would you like to do, co-pilot? Okay. Bridge, this is Nav. Ship move, please. 20 meters, bearing 155 at 0.2 knots. That's correct, thank you.
Say again? Oh. Yeah. Yeah, we're in a good position here.
Interesting, looks like there might be a... I don't know, why is there a... Maybe there's a dip to your port side?
I think so.
Yeah.
Face it. I'm on a little step here.
Yeah, there's not a lot of colonization, at least not evident.
That was fast.
All these rocks.
Yeah, I see it drops off.
Yeah, it drops off, okay. Yeah.
It might be just small animals in those cracks that we are not able to see from a distance. Maybe pilots, we can take a quick snap zoom and some of the rocks on the left to see if there's anything living among the crevices.
This is a nice little sandy step to take a rest. Okay, video. You zoom in.
Take a look at it. Let's see if we find any. Oh, there's a sponge.
Huh. Oh, is this similar to what we saw below? The come around from a different angle. The George O'Keeffe sponge.
It does look similar to me. You see this, likely to see a tube, maybe a worm or something else in the top right corner of the screen. There's a barnacle on the right.
Yeah, this looks like it's the same sponge, Santiago, another one of these polyopagons.
Okay, if you keep the zoom video, I'll tilt up.
Matt is becoming a sponge expert here too.
It should come out a bit so we can see more.
Hardly. Although he's renaming a lot of species here too. So this looks a lot more encrusted with iron manganese here. As you see, much more rounded edges on the rocks, not so angular.
Yeah, this still looks like some kind of volcanic breccia to me.
Yeah, it doesn't look like there's much colonization here. Wonder if this is an area that was much more covered in sediment recently, and it's just there was a slide of a lot of the sediment material down slope, and now it's just becoming exposed. So there isn't a lot of colonization of sessile organisms.
Okay, you can go wide video.
Sessile organisms are the animals. Doesn't necessarily have to be animals, but here in the deep sea, most of the things that you see are animals. There are no plants at all at these depths. There's not enough light for photosynthesis at 2,900 meters. In fact, there's not enough light for photosynthesis practically zero below 300 meters deep. So 300 meters is approximately 900 feet. Right now we are about 9,000 feet deep below the sea surface.
May or may not be some kind of gap
sponge stock up above.
Yeah, it looks like it could be.
There's not really much on
zoom here video.
Serious either.
Yeah, there's something small sticking up.
Little coral there in the lower left.
Yeah, that could be a small octocoral whip in the left. I see some structures that resemble polyps protruding from the center axis.
An even smaller one there, further to the left.
That looks like the primnoid octocoral that we've seen as a large whip, or also as a branching coral. We saw a few of these earlier in the dive. When we zoom in the base, you could see there was a broken off piece from a branch. It's a very small one, indicating that it's not as old as the ones that we observed deeper.
Whenever you're clear video, you can go wide. Yeah, look at my sonar. There's something big across the gap.
Yeah.
Yeah. Any interest in this stock sponge here?
It does look very similar to the one we just observed, but there's not much else around here, so we can do a snap zoom, and maybe along the way we'll also see something else.
Yeah, that move should be underway, and it'll just last 2 or 3 minutes. 20 meters.
And if you look up the screen in the back, in the
I think so.
Ahead, it seems like we're gonna see another change in terrain soon.
Good copy, Bridge. Yeah, we see about a 5 or 6 minute delay at this depth.
Yeah, so this looks to me like another Califactus glass sponge. Likely the same kind we just imaged with the slender, long stalk.
Could we take a quick look at how it's attached?
Okay,
Absolutely, Chris. Let's take a look at the base.
Yeah, good enough. Just checking something. Thanks very much.
Thanks. So, Chris, can you tell us what you look at when you, or what characteristics
Alright, we can go wide video.
you do identify when you look at the attachment of these sponges? That will help you identify what species or genus it may be?
Yeah, so some of the important clues to figuring out what some of these sponges are one, how it's attached to the substrate, two, whether it has a stalk or not, three, what's the nature of that stalk? Is it very fibrous or is it very solid? And four, how does the stalk attach to the body of the sponge at the top? So that particular sponge, the attachment was sort of gluing it to the substrate rather than fibrous, so that helps you narrow it down a bit. The stalk itself was very solid, so that again rules out more possibilities. And then it was attaching to kind of the back of the sponge where there's sort of an oval depression. You couldn't really see it there, but I could tell since I've seen enough of them. That's very informative. That gets you into the family Roselidae. And then just the nature of the body of it will bring it to Caulophacus. So those are some of the things that I'm trying to look at when identifying the sponges.
Thank you, Chris. It's a great explanation of what
you want
things
another 20 meters, 140. That last
you're
move
looking
should
for
be finished up.
when we zoom in the sponges.
Yeah.
So Chris Kelly, University, Hawaii, is the voice you're hearing. And he's an expert in most things, but especially in sponges.
Another ship move, please. We'd like a move of 20 meters, bearing 140 at 0.2 knots.
You see another octocoral whip, like the same kind of, I see it.
That's correct.
Bamboo whip that we have observed deeper.
Excellent.
So we have moved vertically along this slope about 100 meters so far during this dive. RV Nav, this is watch lead.
Go ahead, watch lead.
Can you give us an update on how much time do we have remaining in the dive?
We have just under an hour left on bottom.
Roger. Thanks. So we have about an hour left on bottom.
How many meters to
it's about 110 meters until we run out of data on HiPAC.
Which is,
so we're not in a rush.
0.3,
yeah.
Yeah, something happened there. I think we can assume it kind of continues up.
Yep.
Seems
thanks, Bridge.
As Matt, you were remarking, is getting steeper here slowly. Although the terrain looks a lot different than it looked down deeper.
Coming back to center.
There's not smooth surfaces or large boulders. Like a lot of those large boulders we saw down below roll down from this area or higher. And also the sediment accumulations that we saw also likely were caused in part by flows of sediment just washing off downslope from these shallower depths on the Seamount. So we're diving on Seamount D. We're still open to suggestions for a name. We would like to give it a Samoan name. And since we'll be calling Seamount D, it would be great to have a name that starts with that letter. And again, we've observed several potential new species throughout this expedition, including several today that we have also collected. And it'll be great, and we'll encourage the scientific community that will take a look at the samples, and if we discover new species, it'll be great to give Samoan names to give recognition to the place we are collecting them.
Hey, Santiago, we were just looking at sediment there a second ago, and there were lots of amphipods, small black things swimming over the sediment. You saw that or not, but it looked like maybe 10 or 20 of them.
Tim, can you...
So you may want to look over there.
I couldn't hear you very well. You sound very, not very loud. I was wondering if you could repeat what you just said.
Yes, Santiago, it's Tim. I don't know if you can hear me or not.
Hear you better now, yeah.
As we were just here imaging it with full far up field, wide shot, there was a lot of dark amphipod-like swimming animals. I can see them now again over the sediment. You see them?
Over the sediment. I don't see them. We can take a closer look. Oh.
As I look, it may be actually in the foreground, but I see things swimming in the water column. Many of them.
Yeah.
Well,
Paulo, can we zoom in? And I think those might be particles that we're washing up, but Tim maybe thought it would be some swimming amphipods in the center of the screen over the patch of sediment. Not right here, but further.
Actually, video, if you go in center screen, there's a little, here you go, hold there.
Oh, there's a swimming Holothurian here. Do you think that may be one of those, Tim? This is a very small sea cucumber.
I think these are sea cucumbers, and sometimes you see them in big swarms.
Oh, really?
Observation, Tim. They look like Holothurian to me.
Yeah, this is definitely a sea cucumber swimming. But I wonder if there were more of these as well. That's what Tim was seeing. I saw also some sediment particles lifting up. But this is a very small one. You see the length of the sea cucumber that is swimming. It's about
co-pilot, I should
1 inch.
Move if you haven't already.
Which are only a couple centimeters. So these are also sea cucumbers like the one we observed earlier that were attached to the surface, particularly the surface of sediments, and we saw that the digestive system was full. You could trace it with the sediments. This one is empty. They usually do empty their guts essentially before they take off and swim. But this is a different kind from the ones we observed earlier, I believe. Although this seems to have a sail as well.
Sure.
Thanks, Bridge. Might be the same kind we saw with a sail that had 4 lobes.
Fast now. Okay, there he goes.
Thanks, Pilot. Maybe we can zoom out and see if there was... Oh, there's a clarodicid carnivorous sponge. Yeah. So also when we zoom out and see if we see more of those sea cucumbers swimming around.
You're interested in a zoom on the sponge?
Yes.
Okay, go in video. I just happened to stop here.
Yeah, so this is a clarodicid carnivorous sponge. It's different from the other glass sponges that we've seen that look more like vases or have the stalk and the round heads. This one actually can prey or eat animals. I think I see a jellyfish perhaps attached to some of the spines. Is that full zoom, pilot?
Yes, I can set up again. I just happen to stop here after the jellyfish. If you come all the way wide video, I'll get a little closer.
Yeah.
It's quite tiny, actually.
Santiago, this is Tim again.
Hi, Tim.
After the zoom, I'll go back in. Or I'll go ahead.
Yes, I think what I was seeing were small amphipods that were swimming over the rocks and over the
Let me
sediment.
take my camera tilt.
They were in small patches, it looked like to me.
Okay, tilt up.
When we zoomed in, then we saw that small Holothurian, which was really fantastic. Anyway, when you zoom back out, you may want to take a look at things that may be swimming near the rocks.
Yeah.
Many of them, I think maybe more than 2 dozen of them. So anyway, hopefully we'll see them again.
Yeah, we'll take a look at that, Tim. And I do think, yeah, this anemone has captured a jellyfish or a ctenophore. It is.
Stirring up too much sediment here.
You can see it
only 20% down.
attached to the those fibers, those spines that are sticking out from the center of the sponge. Yeah, pilots, maybe we can tilt the camera left. We got indications there might be a lot of amphipods or little crustaceans maybe swimming over the rocks.
Don't see anything. We'll do one zoom, then I'll have to move on. Video, you can zoom in. Try to focus on. I think it's just.
Tim, do you see any of areas that may have those amphipods that you were seeing?
Yes, I'm actively looking, and I think they may be more higher in the water column than I.
Higher in the water column?
So I saw a few of them near the sediment, but I think if you zoom out, you may see them cross in front of the camera in the water column.
All right, maybe zoom out and see if we
that
can.
last move is probably
see them.
Finished moving serious now. Looks like there might be a little bit of a steep feature up ahead.
I've seen a lot of particles. I think we're stirring with the
He's about
ROV2.
18 meters up, and he's seeing the top of it. Sure, I believe, yeah, upslope.
All right, I'll keep looking, but I think these
on high pack, so
are clearly swimming. They're taking, they're choosing just their own directions back and forth, you know, and they're dipping like they're amphipods.
Okay, yeah, please give us a shout out when we see them again. I couldn't see them, but we'll keep an eye out for them.
So you get pushed out and see this feature, and then go from there.
Sounds good.
Seems like we're reaching our outcrop here.
Yeah, maybe some phyllobasalts visible at the base.
There is something floating, yeah. That looks like a shrimp. It's a different kind of shrimp from the nematocarcinids, the red nematocarcinids.
Okay, you can keep going in. Frame it up. Up.
Wow.
This might be eggs that they're carrying.
The orange bit attached there?
Yeah, that's what I was thinking, maybe eggs. Wow.
Beautiful swimmers.
Look at the coordination of the legs.
Tighter video.
Tim, were you gonna?
So does anyone know? I don't know mycal crustaceans. Tim, is this a mysid or something, or what is this?
Yeah, Tim, do you have any thoughts on this shrimp?
Yeah, it looks like a mysid to me. And I don't know if those are eggs or not. That could be. That they typically would attach them to the abdomen in the central part of the body. So it's not. They're located in the place where you would find eggs on these guys. But yes, I would call this a mysid shrimp.
Okay, let them swim away. Okay, we can go wide.
They're great swimmers. They spend all their time in the water column.
Wow, look at this outcrop. Looks like we might have some pillow structures, but they're not You compare these with the structures we saw at Bioluminescence that erupted in the last 5 years, and it's a lot harder to see the structures here. The surface has been eroded a bit, and a lot, and covered with ferromanganese.
It looks like
maybe volcanic breccia. It's hard to tell from this distance what we're looking at on the right.
Yeah, there's some more. It looks like a stock, sponge stock. We haven't seen that many dead sponge stocks here compared to previous dives. Philo, can we take a quick zoom at that center stock?
Okay.
Just wanted to see, they usually see several different species attached to these dead stocks.
Okay, hold there.
That normally don't see on the living animals. So you can see there are some barnacles. There's a crinoid attached to it. There's a couple of hydroids, a really large one sticking on the left. There's a tube, making a tube. And at the top there seem to be some zoanthids or anemones. Not sure, either one. And yeah, several hydroids sticking out from the sides and a crinoid.
What's above the crinoid there? Just above it.
The tube?
Yeah.
Yeah, it looks like another hydroid.
Huh.
But there's also some, we've seen some worms, some like serpulid worms, or sabellid worms, more general. Also attaching to the stalks.
Is there another crinoid there?
Collected a couple of these
hydroid?
stalks.
Looks like a hydroid.
The one in the center, you know, it looks like a crinoid on a stalk. The juvenile stage.
Oh, so the juvenile stage are on stalks.
Some of these comatulid crinoids, yeah. So we actually did collect a nice specimen of similar community associated with this dead sponge stalks a few dives ago.
So you're interested in collecting this one?
How's that move progressing?
Yeah, I was looking at my notes. We did collect a stock like this in Utu at a similar depth, and this looks, this small community assemblage on this skeleton of the sponge looks so similar to the one. I see what looked to be the same kind of species attached to it. The white barnacle and a couple of different crinoids and the sabellid worms.
Utu must be 150 to 200 nautical miles northwest of here.
Yeah. So this provides us some evidence that the depth, creating the depth profiles of the depth distribution is maybe driving the biodiversity patterns more than the distance within this region in some American Samoa. So yeah, that one in the center is a hydroid. It's a solitary hydroid.
Do you want to go in on the black rock in the back? Looks like there's lots of things living on it. Just to the right. Do you have any more zoom there?
There we go.
Back there.
We're gonna get a good look of this crinoid that is on a stalk. So yeah. So this looks like we collected one of these at I believe it might be the same species. You see it has a stalk, but actually when we got it, I got it in a bucket and actually detached from the stalk. And we know that these comatulid crinoids, the ones that don't have stalks, have a development stage where they do have these small stalks. We collected several of these stalks as well on dead coral sponge. So it might be an indication that these dead coral stalks may be an important habitat for the development of crinoids. I don't recall seeing these small stalk crinoids on rocks, although they're so small it might be hard to observe from the distance on rocks. Yeah.
Okay, there's a shot with lasers. Ready to move on, Watchley?
Yeah, thank you. That was really great to be able to observe those associated communities on these dead sponge stalks.
So Andy, for that, for a move coming up, might need to have you turn to starboard.
Oh yeah, just going that way.
Yeah.
Okay, I'll come around too.
There's another of those whip corals, bamboo whip corals.
We could try, yeah, try like a 150. What do you think?
There's a small sponge under that boulder right by the lasers. I don't know if we could take a quick peek at that.
Sure. Pilot, could we take a, you see the sponge under the center boulder?
On a little white circle?
Yeah,
Or
so right below the lasers.
okay.
Yeah, it's white and more like an oval shape.
Oh, below the, I see the
Yeah,
bottom of
yeah,
the
yeah.
screen there?
The bottom of the screen, correct.
Okay, stand by video.
We'll just wait till he's done here.
Copilot, can you deploy the port lower swing arm? Try moving it around to get some light down here. Then you might have to bring it inboard, yeah. Yeah, that's better. Okay. And then if you get lasers off, we can go in free to zoom video. Yeah, you can try a little. Light looks pretty good though compared to before.
Okay, this is a glass sponge of some kind. I think it's again in the family Euplectellidae, and it could be in the subfamily Corbitellinae, and that's about as far as I can go with this one.
You see again that what looks to be a small translucent opheroid that we've seen on sponges and corals throughout the dives. And you can see here the detail of also the manganese, iron manganese crust on these rocks. The texture is characteristic of that crust, covering on the underlying basalt rock in this seamount. Thank you, pilot. This is great. We can continue.
Okay, video, once you have your shot, you can go wide. Excellent. Yes, please. Just, yeah, nice and smooth. That was good. Pretty fancy lighting there.
Okay, we could probably put in the move. I was looking at 150, 20 meters.
Sounds good.
Bridge, this is nav. Ship move, please. Range 20 meters, bearing 150 at 0.2 knots. That's correct.
Okay. So how far to waypoint 3 now? Off the map was close.
Waypoint 2.
Sorry, waypoint 2, yeah. What depth is that at?
213 meters. Hard to give you a depth there.
Okay, coming back to center, co-pilot. Coming back to
Thanks,
center.
Bridge. Somewhere around 2,800 meters. Be waypoint 2.
So we are at depth of 2,875 meters.
Your video.
There's an anemone attached to the rock. Right now we're zooming in.
Okay, hold it there. Watch lead, any interest in this anemone?
We just spend a couple or 2-3 seconds imaging and we can continue.
Coming up. And there's a rock.
It's very small.
Okay, you can go wide video. Seems like you either set up fully and do it, or you just don't do it.
Yep. I agree. Watch your port side.
Yep. See it there, I'm serious.
There's some more boulders here, large boulders. There is something attached to the boulder here in the center.
What do you see attached to it?
Pilot, you see the bottom of the screen? There's some tentacles.
Oh yeah.
Not sure what that
Very
is.
small. Bottom left?
Bottom center right now. There it is.
I see. Yeah. Okay, video, you can go in. Crinoid. Okay, hold there.
Oh, it's a crinoid. Yeah. Haven't seen. This is the first one we see attached to the rock in this dive. It looks similar to the ones we observed clinging to the dead glass sponge skeleton. Thank you, pilot. We can.
No problem. You can go wide video.
Maybe another steep feature coming up.
Okay. Okay.
Yeah. Yeah. There he is.
So there's some more outcropping boulders of medium and small size here. less abundant sessile fauna attached to this. In fact, I think the highest abundance of corals and sponges we saw was just as we started the dive. There was a couple of really large boulders. They had many coral colonies and sponge colonies of several species attached to them.
Okay. So that move was called in at 127.
So you've got some type of coral that's center upper, just above the center of the screen, I guess.
Yeah, I see it.
That's what it looks like.
20 meters, and I'm stretched out.
Pilot, do you see
and
that?
It's probably just going to start hitting you now.
Chris, you're talking the one in the center of the screen or the top left corner?
I think he was just...
I think there's something in the center of the screen, but there's also something upper center, too.
Yeah, I think that's the same thing I saw. So
7 minutes of bottom time. 27.
Roger, thanks. We have.
So we're gonna look at this coral.
Yes, please.
Stocked creature here.
Yeah, we have a little less than half an hour left on the dive. So this looks more like a dead sponge stock that is just the fibers are getting separated or pull apart.
Yeah, I agree. We probably don't need to spend much time here, but that's what it looks like to me, too.
Yeah, that's good, Pilot.
Okay, let's zoom out. That last move was toward 150, I believe.
Yep. And there was something else on the left top. I just had to tilt the camera a bit towards the left.
I'm sorry, you wanted to look toward the left?
Yes, please.
Okay, you got it.
There's microcosynid shrimp on the right. We saw another coral on the top of the rock here on the left. So there it is. It's a small whip. I believe it's the same kind we've seen throughout the dive. We can do a snap zoom to confirm.
Sure. Let's go and zoom in. Hold there.
If you could zoom in towards the bay. Oh, yes, yeah. It's the same kind, the same bamboo. You can still see the nodes. They're not very frequent, but I see a few of the nodes. All right, thank you, Pilot. We can continue.
Go ahead and zoom out. And you'll only be moving for about 3 minutes, co-pilot. Yeah, it looks like it.
Oh, yeah,
There's another Caliphacus.
I think it's right here.
Sponge on the left. Further to the left is something else in the background. You see that, Pilot?
Yes, so I will move over there. Looks different.
Yeah, it is different. I agree.
Go ahead and zoom in, and I'll keep moving closer. Hold there.
So this looks like a sponge, but it has two stalks.
Oh. Let's zoom out video. I'm gonna come over.
Yeah, I think this is the Ninja Mutant Sponge. That is
that a
one
scientific name?
bizarre Caliphacus, I guess, but I have never seen one do that before.
Yeah, you see the stalk branching and then coming together again at the head. That's really unusual.
Go tighter.
So, Chris, do you think this will be an interesting collection? Do you think this is
I think it's a mutant.
A mutant?
I don't really think it's a different species.
Not representative.
But I mean, who knows? I think there, you know, maybe if we can find something else in the next few minutes before the dive ends, that might be better.
Okay, sounds good.
Okay,
let's continue.
Looks like starboard outer is the last remaining box.
Just going to peek over at the port side here. See a jagged rock feature.
So you guys feel like you're done moving?
Yeah, it should be done.
Would thinking ready for another one? Or not?
Yeah,
Wait.
we can do, well...
We can wait, no rush.
Yeah, let's wait a couple more minutes.
Yeah, there's some steep features here.
A big outcrop like this in front of us would be a good opportunity to find some additional biological
Yeah, we'll
specimens.
see what lives on here. Seems like a good outcrop for sessile organisms to
Again,
attach to.
This looks more like a block that's fallen from above rather than a... Oh,
Yeah.
there's a sponge there in front of us.
Yeah, that's like... There's another couple of sponges on the top, stock sponges. We've got a Holothurian in the back. The one in the foreground is another of those bamboo whips.
No interest in there, in any of these?
I think we can continue, yeah.
Okay. I'm going to come back over
Well, the
to
bamboo
starboard
whip
here.
would be representative.
Yeah, we got one already.
Oh.
That's the one
That's
with the
right,
rock.
That's right, that's right. With the fish rock.
Yeah, my only concern, Nev, is we got to move
Continue ascending. Have around.
Maybe let's do a 10-meter move.
Close to 20 minutes left of
we're
this
almost
dive.
certainly going to sample in the next 20 minutes. So.
Copy that.
Depth of
It's about 10 minutes between when we call
2,861
it in and when it
meters.
stops for Sirius.
Bridge RV nav.
Oh, this does look like an area
Ready
of
for a move.
landslides. And
10 meters, bearing 150
Yeah, it's
degrees.
just constant
speed decimal
debris
two.
between
Just peek over to my starboard side.
outcrops.
It's a good
The
copy.
outcrops are false outcrops. They look to be extremely large blocks, maybe 5 to 10 meters or even larger, that have rolled downhill. Like this right in front of us, maybe. It's hard to tell, but
so this seamount rises from the surrounding seafloor by more than 3 miles. So it's a really large
Move initiated. Thank you, Bridge.
underwater mountain. There's a small fraction of any of these features that we can investigate in a single dive.
Yeah, 15 minutes, pilots. Do we got to pull off bottom?
So we have 15 minutes left of the dive. Just a couple more of those small bamboo whips.
Fish off in the... Is it a fish or...
There's some larger
Maybe not.
outcrops coming in front, coming ahead of us. That's a definition of a seamount, is any feature that rises more than 1,000 meters from the surrounding seafloor. And there's tens of thousands of these seamounts underwater. And they can form by different processes, different geological processes. Some can form at spreading centers where two tectonic plates are coming apart. Also can form in hotspot areas where there's a great source of heat coming from the inner earth deep in the mantle, reaching the crust, melting the oceanic crust and producing volcanoes and seamounts like this one. This is an example of, very likely an example of a seamount formed by this hotspot process. Well, it's not a lot of attached fauna on these rocks.
Yeah, what's wrong with these rocks? They ought to have plenty of biology on them.
I know.
They should.
Disappointing rocks.
Move complete.
They're
Take a
bad
picture.
rocks.
Bad
rocks.
I don't even see any fish rocks or shadowfish.
Possibly a small sea star to the right center.
To the right center. Oh, I see him.
I don't even see it yet.
Go ahead and zoom in. Might just be a
It may not be
sediment
anything, but I think it's a small sea star. No, it is a sediment star.
That's so. Is that related to the fish rock?
Yeah, I think so. We've been deceived several times by the geology that has some biological shapes from the distance. Spider, can we take a look at that small coral that is sticking out of that rock on the top of this?
Sure.
Oh, this looks like a flat plate. I don't know how to describe this morphology. Like a flat rocks here. See a swimming polychaete. Right here in the center.
Okay, go ahead and zoom in real quick. We'll sit down if we need to. Looks like a dead sponge.
Yeah, it looks like a dead sponge.
Huh.
Yeah, we can continue.
Okay, zoom out, please.
Okay, I called the move in 5 minutes ago, so Sirius said start swinging 10 meters.
Okay.
See something white perching on those rocks in the center, in the back.
A little, almost like a dust devil on the seafloor in front of us.
Oh yeah.
Zoom in.
So sort of
Zoom in.
family of squat lobsters. Sorry, not a family. It's a single squat lobster, but I thought there were 2 or 3. This is one large one.
Well, I'm not so sure this is a squat lobster. Yeah, I guess it is. It's just a really big one, huh?
Yeah.
Zoom out a bit.
How big is that?
I can zoom out with laser, so it's about 10 centimeters, maybe 15 centimeters
Zoom out a
from
bit more.
the tips.
Is it possible to collect one of these?
They will run away or swim away.
It's like our claw is bigger than yours.
These are really hard to collect with this. You usually need a slurp gun to collect the squat lobster.
And they would just hold on to the jaw.
Unless it really attaches to a coral as an associate, but this species we normally see moving around on their own.
Well, this is a squat lobster in the family Munidopsidae, and it's a genus Munidopsis. We were just coming up on it head on, so I couldn't really recognize it immediately, but you can see the big central rostral spine coming off of it and the short stubby claws that characterize the Munidopsis.
Yeah.
This is the first one we've seen today, right?
It is.
Yeah, that's correct, Tim, as far as I know.
Yeah, that's what I have. Okay.
Yeah.
Sure.
Okay, zoom out.
Okay. There's some more large boulders here and more just sediment with ripples in between them. Again, indicative of currents, and I think we're seeing a current starting to pick up here. This looks like a perfectly good habitat for corals or sponges, but they're just bare rocks. Again, we might be seeing
sponge.
Oh, there's a sponge, but just one.
One lonely sponge.
There's another one at the top of the screen.
Yeah, this looks different.
RV nav. Go ahead. How much time do we have remaining down here?
Hold there.
7 minutes.
We have. Thank you. We have 7 minutes remaining on bottom today. Got a little coral back here. Is that a coral?
It's a sponge.
This is unusual looking.
It looks like one of those sponges that Chris was mentioning earlier as a demospongia rather than a glass sponge.
Yeah, I agree, Santiago. This looks like that thing that we call Stellodorix, or at least it's in the family of Vulcanellidae, demosponge.
Go ahead and zoom out, please.
Is there any interest in collecting this,
Yeah, I was
Chris?
going to ask you that, Chris. We only have a few minutes left in the bottom, and we're not seeing a lot of animals here, and wondering if you think it would be a good sample to collect to characterize the sponges at these depths, or at least a representative of the sponge communities. Either this one, this, and this Califacus on the right.
Is that going to be a difficult collection, or because it's kind of on the side of that big boulder? I'm just curious how this would work.
Yeah, that one might be difficult. We can ask the pilots. But now there was a Califacus sponge. We just passed it. It's just probably it's under us right now.
But this one would be of more interest than the Califacus, even though I'm not a demosponge guy. But these, I think Shirley and Rob and some others would be quite interested if you collected this one.
I think we can get it.
Pilots is confident that it can be collected, so I think we can give it a try. I think it will be important to give it also a chance to the sponges to be characterized in more detail in addition to the corals we observed to the dive. They seem to be the two main characters at these depths.
I'd really like to consider collecting the rock that's attached to that sponge. We could.
That one might be a little too big. Probably set a record though.
Just keep it on the claw as we ascend.
Yeah, just have the claw hold it close to the ROV as we ascend, and
starboard outer is available.
When I saw the laser beams next to the sponge, I realize I greatly overestimated its size. Those beams are only 10 centimeters apart, so that sponge is just a couple of inches tall.
Yeah, it's a small one.
You know, it's kind of interesting to see one of these things out in the open because this particular type of sponge is always concerned about being stalked. You got that joke, right?
Oh my God, is it getting that late over there in Hawaii?
Yep.
Is this going to be brittle or?
Let me ask Chris. Do you think this is going to be soft body sponge or more brittle?
It's not going to be brittle. It should come off fine, and it won't come apart.
Okay, that's good. So I can get it in the jaws and lift up.
Great.
And
yeah, that should be easier to collect then.
Yeah. Okay, hydraulics up. Yeah, I'll have to grab it and then come up and...
You know, Tim, here in Hawaii, we complain if the temperature drops to all of 60 or 65, and likewise, we complain if the time drops to all of 4 p.m. in comparison to the East Coast, which is, you know, much later at night.
Yeah, I assume you're watching the tides and the sets roll in too, and then you have to bolt out of there to catch them.
Yeah.
Oh, nice collection there. You got it?
Nope.
Thank you, Santiago.
Okay, gonna come up off this rock.
Starboard outer pilot is open, available.
Yeah, just so I don't float around too much. Okay. Yep.
So this demo sponge is going into the outboard, starboard bio box. This is specimen 7 of the dive. Biological specimen.
And with that, it's time to get off bottom, pilots.
All right, let me stow the craft here properly.
So this is likely the end of the dive. Today's dive on Seamount D. We started at a depth of 3,000 meters and we are ending at a depth of 2,854 meters. We moved over really steep terrain. There was really interesting diversity of substrate. There was large detached boulders with colonization of several species of corals and sponges on them. As we move on later, we also saw some sedimented slopes with ripples indicating currents.
So let's just
in this area
kind of come up in formation for.
But then we encountered areas of smaller cobbles, as well as area of much chaotic morphology in the seafloor. We ended up in spot where there's several outcrops and some sedimented slopes again, but not much colonization of the rocks towards the end of the dive, which is interesting. And then maybe due to recurrent
why don't you start coming up a bit?
Landslides that might happen in this area given the slopes and the evidence we see for a lot of material falling from shallower portions of the seamount.
If you want to go ahead and tilt down. Just keep coming up. So you can turn and line up with the ship. You need to come to port
before
like 30 degrees or so.
We lose everybody in the line. I would like to ask if anybody wants to go over the plan for tomorrow. We had a discussion and we picked a dive track earlier before this dive. We'll be happy to bring it up again if that's something that anybody wants to discuss further. So just let us know now and we can set that up within the next few minutes.
And just go ahead and point your camera straight down. Good. Thank you.
Yeah, this is Tim. I really like the dive track or the compromise that you guys came up with before this dive. I thought it was pretty well conceived. As long as Matt feels good about it. Sorry, the biologist felt good about the slope and hitting the ridge and making sure we have enough time on the ridge. But as long as it covers the geological goals, I think it's a good dive plan for tomorrow.
Great. Thanks, Tim. Yeah.
Dive is fine with me.
Yeah. Seemed like you covered all the goals and objectives for the dive tomorrow.
Did you hit the SCS timestamp?
Yeah, and ditto from me, all of Tim's comments as well. I would agree. Completely very satisfied with the final plan.
Great. We'd like to hear that. I don't know if anybody else in the line has any other thoughts about going over the plan. If not, I think we can start saying goodbye for today's dive. It was really interesting to see the comparison at the same depths or similar depths to the depths we dove on Seamount, the Utu Seamount the shallow dive we did there, relatively shallow, 3,000 meters, but that was the top of that seamount. We observed similarities in the communities, at least in the species, the kinds of species that we observe here, and we made new valuable observations and collections that will help us further constrain the diversity in this area and at these depths. And we look forward to continuing the next 3 dives that we have. We're going to be at shallower depths than today. Actually, 2 dives left we have. We will be diving tomorrow another seamount. We're going to target the summit of a seamount, the Malulu Seamount. It will be at the depth of around 2,500 meters. We expect to see much greater abundance of coral and sponge communities, as well as characterize the geology of the
let's
seamount.
come up a bit right before we start coming ahead, yeah.
And we are going to have our last dive of the expedition the following day. That was originally planned to be the first site of diving on a Nautilus community. There was a great interest in not losing this dive. The original one we had a malfunction of the remotely operated vehicle, and we had to abort the dive while the vehicle was in the water. So we're going to have the opportunity to go back to the site and do the dive that we had or planned for as the first dive of this expedition. So that should also be a really interesting dive at shallower depths, around 500 meters deep. It will be an excellent depth comparison to the faunas we see at different depths. So we're going the last 2, including today's dive, and going forward the next 2 dives, we're going to from deep to shallow. So it should be really interesting, and we're excited for the observations and discoveries we'll make in the last 2 dives of this cruise.
Hi, Santiago. It's Amanda.
Hi, hello. Can you repeat that? Sorry, I didn't hear you very well.
Oh, this is Amanda Nepburn.
Oh, hi, Amanda. How are you?
Good. How are you doing?
Good, thank you.
Sorry, I was having a little trouble calling in, but I did want to just go over the midwater portion of tomorrow's dive.
Sure.
I think we have it pretty...
Sorry, go ahead.
Yeah. So I think we have it pretty sorted out, but Kelly said that
Go ahead, Bridge.
you were going to request a dive extension.
Yep.
Yeah, that's
yeah,
that's the plan. So we are looking to have an extended dive, extending it by 2 hours tomorrow to accommodate for the midwater transits. But in fact, if you want to stand by for a couple of minutes, Kelly just stepped in the room, and we're gonna bring in the lead of the ROV team so we can discuss the specifics for those transits. So if you stand by for a few minutes, we'll get settled here to have that discussion.
Sounds good. Thanks.
So we just got Carl here, the leader of the ROV team, and Kelly's getting connected as well. We're waiting on Mimi to bring up the maps for tomorrow's site, Amanda. So it'll just be a few more moments.
no more than 2,700. That's what waypoint 2 is, and that's way up here. Yeah. I'll zoom out just to discount double check. Yeah, we do have. Yeah.
Okay, so
there is missing data here, so I mean, the streaming looks good. You're pushing out, so I'd be okay with even going up, maybe 2,600 and then scooting over. I don't see why not.
Okay. Coming up 30 meters per minute. Winch looks good. Shiv looks good. Streaming looks okay. I'll push a little harder forward here.
Yeah, we'll get you guys to 2,600, then I'll go half an hour over ground.
Or let's just do we'll do 0.3 over ground for now, just because so much wire out. I don't want to get, you know,
okay,
half a kilometer of layback or something. You know. All right, thank you.
okay, Amanda, we have everybody now in the room connected, and we
can only guess.
If you look at the stream tree,
right. So I'd say 2,700.
You should be able to see the Google Maps.
You know.
Yeah, with the general area.
I've never seen contours cross like this before.
Yeah.
For tomorrow's dive
situation.
So Mimi's just pulling the, trying to get a line to measure distance between today's dive site and tomorrow's. You see it there in yellow.
Yep. Was this freshly?
Okay.
Yeah, so we're gonna get there a
Probably
little
fresh data. Just got it.
early, hopefully, and we should be able to do one extra line.
Okay, yeah. You guys are
mapping
now
data
2,700.
over
I'd be
the
okay with
Seamount
going
in the morning.
decimal 3 over ground.
And
Okay, let's do it.
You guys are.
Come to the
Bridge
summit,
RV nav.
and on the way over
Yeah, we'd like to
to
come
Seamount, we
ahead
have a little data gap
decimal 3 knots over ground.
So as long as we can go good transit speed, we can get that done.
Oh,
So
let's keep it
pull
at
up
30, 31.
The site.
Copy that.
Got a little more room to play here.
Thank you, Bridge.
And we talked about this one this morning. It seems like a good mix between bio
Down through the water
and
column, just
geo
so it's in the lake pool and we can
objectives
see
in terms of climbing
something
a
that swims
slope
by.
and going along a flat.
Right. So Mimi, can you maybe zoom in on the dive track? Because
Like
I don't
50
think
meters, yeah.
Amanda, did you follow the discussion this morning, or?
I wasn't on the discussion.
Okay, yeah. So
but
just
Kelly had emailed me that it's basically at the end of the dive. The plan is to be on the summit of this.
yeah. So you can see in the image the top of the seamount. So the colors indicate slope. So it has the idea is to come up on one of the sides of the seamount. We might do a little bit shallower at the starting point to get more time on the top of the seamount. So the idea as we come on the slope is to be able to collect some nicely attached pieces of rock that we can sample to characterize the geology of the seamount, and then look at the